Hello there, and welcome to Biology with Charles, where curiosity gets cozy and science takes the scenic route to slumber. Tonight, we are turning our attention to a world so small, so busy, and so close to you that it has been with you through every meal, every illness, every quiet evening, and every ordinary day without asking for applause. We are talking about gut bacteria, the tiny residents living mostly in your intestines, working through the dark like a night shift so organised that even the most devoted office manager would have to admit defeat. They do not wear uniforms, they do not send reports, and they are not especially concerned with your plans for tomorrow, yet they help shape digestion, immunity, mood, and the general rhythm of being alive. It is a strange thing really to realise that part of what feels like you is also the work of countless microscopic organisms going about their business in silence. Before we settle into this hidden inner landscape, I would love to know where in the world you're listening from tonight and what time it is in your little corner of it. There is something fitting about that, I think. A calm video about the unseen life inside the body, arriving in different places at different hours, while all of us carry these same invisible companions within us. And if you find this soothing, helpful, or simply a nice way to unwind after a long day, I would be very grateful if you gave the video a like and maybe subscribed. It helps this quiet corner of science reach more people who might enjoy drifting off while learning about the remarkable things their bodies are doing when nobody's paying attention. For now, get comfortable, let the day loosen its grip a little, and come with me into the gut, where whole communities live, cooperate, compete, build, repair, and quietly help make you what you are. Most people grow up thinking of bacteria in one of two ways. Either they are germs that make you sick, or they are something you hear about in yogurt commercials and then forget 5 minutes later. But the truth is much more interesting, and also much less dramatic than the word germ tends to suggest. Bacteria are everywhere. They live in soil, in oceans, on plants, on animals, on your skin, in your mouth, and in astonishing numbers inside your digestive tract. The human body is not a sealed and solitary machine. It is more like a living environment, a place where human cells and microbial cells share space, exchange chemicals, and affect one another constantly. Nowhere is that relationship more important than in the gut. When scientists talk about the gut microbiome, they are referring to the vast collection of microorganisms living in the digestive system, especially the large intestine. That includes bacteria, but also viruses, fungi, and other tiny life forms. Still, bacteria are the best known and the most studied members of this inner community, so they often take centre stage. There are trillions of them, not billions, which already sounds like too many to count on a lazy afternoon, but trillions. They are made up of many different species, and those species do different jobs. Some help break down food your own body cannot digest by itself. Some help make vitamins. Some help keep harmful microbes from gaining a foothold. Some help train the immune system, so it does not attack everything in sight like an overexcited guard dog. This means your gut is not just a tube that receives food and pushes it along. It is also an ecosystem. And like any ecosystem, it depends on balance. A forest cannot function well if one species takes over everything and crowds out the rest. A coral reef cannot stay healthy if its relationships break down. The same is true in the gut. Health often depends not on having one perfect bacterium doing one magical thing, but on having a broad, stable, well-fed community working together. That point matters because people are often tempted by simple stories. One strain is the hero, another is the villain. Eat this one food and everything improves by sunrise. But biology is usually not that tidy. In reality, gut bacteria are not a neat list of good and bad characters in a children's book. They are more like a crowded city filled with specialists, labourers, opportunists, security staff, recyclers, and the occasional troublemaker. A given bacterium may be helpful in one context and less helpful in another. A species that is harmless at low levels may become a problem if the environment changes and it grows too much. Even microbes that are often called friendly are not acting out of kindness. They are surviving, feeding, multiplying, and in the process they often help us too. So how do these bacteria get there in the first place? You're not born with the fully developed gut microbiome of an adult. Early life matters a great deal. Birth, feeding, surroundings, contact with caregivers, diet, illness, antibiotic use, and the passing months and years all help shape which microbes settle in and which ones do not. The gut microbiome begins forming early and changes over time. During infancy it is less diverse. During childhood it expands. In adulthood it becomes more stable, though never fixed. Throughout life it responds to what you eat, how you live, what medications you take, and what your body is dealing with. That idea alone is quietly remarkable. Inside you is something alive and changing that responds to your habits. A meal rich in plant fibre can encourage some bacteria to thrive. Repeated courses of antibiotics can shrink certain populations and allow others to surge. Long periods of stress can affect gut movement, immune function, and the environment these bacteria live in. Poor sleep, illness, travel, infection, and age can all leave their mark. The microbiome is not like eye colour, settled early and left mostly alone. It is more like a garden that responds to weather, care, neglect, and whatever happens to blow in. If this sounds slightly unfair, that is because it is. Your body is trying to run an intestine, digest food, absorb nutrients, regulate immune responses, keep harmful organisms under control, and somehow also manage a microbial population the size of a small cosmic census. It is rather a lot to ask of one digestive tract, and yet most of the time it works well enough that you do not notice it at all. That may be one of the most impressive things about biology. The systems doing the most constant work are often the ones least likely to draw attention when they're going well. To understand why gut bacteria matter so much, it helps to begin with the limitation of the human body itself. We are not able to digest everything we eat using our own enzymes. Certain fibres, especially many complex carbohydrates found in plants, resist digestion in the upper digestive tract. They pass through the stomach and small intestine largely intact and reach the large intestine, where bacteria take over. There, many species ferment these fibres and produce useful compounds including short-chain fatty acids such as acetate, propionate, and butyrate. These compounds are not minor leftovers. They can nourish cells lining the colon, support the gut barrier, affect inflammation, and even influence metabolism beyond the gut itself. This is one reason fibre matters so much. It is not just roughage, not just some old-fashioned nutritional virtue your vegetables are proud of. Fibre is also food for certain gut bacteria. When you eat a variety of plant foods, you're not only feeding yourself, you're feeding the community that helps process what you eat. That community in return produces molecules your body can use. There is a relationship here, a real biological exchange. You supply material, they break it down. Both sides benefit, but digestion is only one part of the story. Gut bacteria also help shape the immune system. In fact, a large portion of the body's immune activity is connected to the gut. This makes sense when you think about it. The digestive tract is one of the main interfaces between the body and the outside world. Food, water, microbes, and all sorts of foreign substances pass through it. The body needs to be able to distinguish between what should be tolerated and what should be attacked. That is not easy. An immune system that reacts too little leaves you vulnerable to infection. One that reacts too much may create chronic inflammation or misfire against harmless things. Gut bacteria help teach this balance. Early in life and throughout life, they interact with immune cells and help influence how the immune system matures and responds. Some bacterial products calm inflammation, some encourage protective mucus production. Some strengthen the barrier formed by intestinal cells so that unwanted substances are less likely to pass through. In other words, the microbiome is not just sitting there hoping for crumbs, it is part of the negotiation between the body and the outside world. There is also the matter of competition. Harmful microbes do not enter an empty hall and find a reserved seat waiting for them. In a healthy gut, they encounter crowded conditions. Resident bacteria compete for space and nutrients. Some produce substances that make it harder for rivals to grow. Some help maintain an environment that does not favour invaders. This is one reason a healthy microbiome can protect against infection. When that balance is disturbed, after illness or antibiotics, for example, gaps can open up. And as with any poorly supervised environment, something unpleasant may decide it is a very good time to move in. Then there is the part of the story that has captured a great deal of attention in recent years, the connection between the gut and the brain. This does not mean your intestines are secretly writing your diary or making all your decisions while you take the blame. It means the gut and brain are linked by nerves, hormones, immune signals, and microbial products. Some gut bacteria are involved in the production or regulation of chemicals that affect the nervous system. Some influence inflammation, which can in turn affect how the brain functions. Some may be linked, directly or indirectly, to mood, stress responses, appetite, and sleep. This area of research is still developing, and it is wise to be careful. Not every exciting headline about gut bacteria and mental health turns out to mean what it first seems to mean. Biology is complicated, human studies are messy, and many effects are subtle. Still, the broad picture is becoming hard to ignore. The gut is not isolated from the rest of you. What happens there can echo elsewhere. A calm and healthy intestinal environment may support more than digestion alone. A disturbed one may do more than cause stomach discomfort. That begins to explain why the topic of gut bacteria has become so important. It is not because microbes suddenly appeared one day and asked for better publicity. It is because science has gradually realised that these organisms are woven into systems we used to think were mostly human affairs. Digestion, immunity, metabolism, the integrity of the gut lining, and perhaps aspects of mental state all appear to be influenced by this inner ecosystem. The body is less independent than we once imagined. We are, in a very literal way, living with help. Of course, once people hear that, they often become eager to classify everything. Which bacteria are good? Which are bad? Which should be increased immediately? Which should be evicted without notice? But that is where we need a little patience. Many kinds of bacteria live in the gut, and they do not divide neatly into saints and villains. Broadly speaking, some groups are often associated with health when present in healthy balance, especially those that help digest fibre, produce beneficial compounds, and support the gut barrier. Others can become harmful if they grow too much, especially if the system is already disrupted. Some are called commensal, which means they live with us without obvious harm and often with benefit. Some are mutualistic, meaning both sides benefit. Some are opportunistic, behaving harmlessly in one setting and causing trouble in another. Think of it like a town. You want farmers, cleaners, repair crews, transport workers, and people who keep the water running. You do not want the sewer system to collapse or one loud fool with too much confidence to take over the town square. Yet the town is not defined by one person. It works because many roles are filled at once. The gut is similar. Diversity matters, balance matters, stability matters, and context matters more than people often realise. This is also why it is not very useful to chase one miracle food or one miracle supplement while ignoring everything else. Gut bacteria respond to patterns. They respond to the foods you eat regularly, not just to one noble salad eaten on a Tuesday, while the rest of the week is spent in quiet conflict with vegetables. They respond to sleep, stress, medication use, movement, illness, and the general conditions of life. If you want to support the microbiome, you usually do it the same way you support many parts of the body, with consistency rather than drama. Now, because this is only the beginning of our journey into this topic, it is enough tonight to become familiar with the basic truth that your gut is inhabited, active, and important in ways that older science only partly understood. Inside the large intestine especially, bacteria are feeding, fermenting, signalling, competing, and cooperating. They help turn leftovers into useful molecules, They help protect space from harmful invaders. They help train immune responses. They help maintain the lining that separates the contents of the gut from the rest of the body. And they are not all the same. Some specialize in breaking down certain fibers. Some help generate vitamins. Some are tied to resilient gut communities. Some thrive when diets become narrow or the environment becomes inflamed. Some are more abundant in one person than another. Each microbiome is shaped by life history, habits, and circumstance. That individuality is worth pausing on. There is no single perfect microbiome shared by all healthy people. There are patterns associated with health, yes, but there is also variation. Two people may both be well, and yet carry somewhat different microbial profiles. This makes sense because bodies are not identical, diets are not identical, environments are not identical, and life is not conducted under laboratory conditions no matter how much some nutritional advice seems to imagine otherwise. The microbiome is personal, it is biological, but it is also biographical. It reflects where you have been as well as what you are. And perhaps that is one reason the subject feels so absorbing. It changes the way we think about the self. You are one organism, of course. You're not just a crowd in a coat pretending to be a person. But you're also host to an immense population of microscopic life that contributes to the conditions under which your body operates. That does not diminish you. If anything, it makes the body more interesting. It reminds us that life is often cooperative, layered, and less lonely than it appears from the outside. In the years ahead, research will continue to refine what we know. Some claims will prove too simple, some early ideas will be revised. That is the normal way of science. But the central lesson is already clear enough to rest with comfortably tonight. The gut is not empty. It is not passive. And the bacteria living there are not incidental passengers. They are participants in digestion, defense, chemical signaling, and the maintenance of internal balance. To care for the body is, at least in part, to care for the conditions those microbes live in as well. So as you lie there tonight, perhaps already a little more relaxed, it is worth imagining that deep inside the quiet of your abdomen, there is motion without noise and work without ceremony. Fibers are being fermented, cells are being nourished, signals are being exchanged, boundaries are being maintained. Tiny organisms, with no awareness of your plans, your worries, your shopping list, or the song that got stuck in your head this afternoon, are still helping keep a whole hidden system in order. And that hidden system, in turn, is helping keep you going in ways that most of us never think about until something goes wrong or science politely asks us to pay attention. We have only just opened the door on this inner world. There is still much more to sort through, because not all gut bacteria do the same work, and the community inside you is made up of many kinds of settlers with different roles, different strategies, and different effects on the body. Some arrive early, some stay for the long term, and some become especially important depending on what you feed them and how the environment inside the gut begins to take shape. If the gut is a hidden world, then every hidden world has to begin somewhere. It does not simply appear one morning fully built, with busy little neighbourhoods, supply routes, chemical exchanges and microscopic residents, all going about their work as if they had signed a lease long ago. Like any living system, the gut microbiome has a beginning, and that beginning is one of the most fascinating parts of the story. Because before these bacteria become digesters, protectors, vitamin makers, and quiet partners in your health, they first have to arrive. They have to find their way into the body, survive there, and settle into a place that, at first, is still learning how to host them. And so, tonight we turn to the first settlers, the earliest microbial arrivals, and the slow, complicated process by which the human gut becomes home to tiny lives that will help shape so much of what follows. To understand that process, it helps to begin with a very simple idea. Gut bacteria are not random dirt floating around inside you. They are living microorganisms, mostly single-celled, far too small to see without a microscope, yet fully equipped for the business of survival. They eat, divide, compete, cooperate, release chemical byproducts, respond to their surroundings, and adapt to changing conditions. They are alive in every practical sense, even if their lives are very different from ours, and while the word bacteria can still make people think immediately of infection, fever, and public service announcements about washing your hands, most bacteria are not villains waiting dramatically in the wings. Many are harmless, some are helpful, a few are harmful in the wrong place or in the wrong numbers. The important thing is not the label alone, but the context. Inside the human gut, bacteria occupy a world that is warm, moist, nutrient-rich, and constantly in motion. Food passes through, immune cells stand watch, mucus lines the intestinal surface. Oxygen is limited in many regions, especially deep in the large intestine, which means the bacteria that thrive there tend to be well suited to low oxygen conditions. The gut is not a smooth, empty tunnel. It is a landscape full of folds, gradients, shifting chemistry, and changing supplies. Different parts of the digestive tract suit different microbes. The stomach is acidic and hostile. The small intestine moves quickly and contains digestive enzymes and bile. The large intestine is slower, denser, and far more welcoming to the long-term residents that make up most of the gut microbiome. For a long time, people tended to imagine the body as a sort of personal fortress. You are you, the outside world is outside, and anything foreign that gets in is probably bad news. Biology, as it often does, turns out to be less tidy than that. The human body is better understood as a living environment. It has borders, yes, but also surfaces, openings, exchanges, negotiations, and relationships. Microbes are part of that story from very early on. In fact, one of the most important events in early life is not something you can watch without a microscope at all. It is the beginning of colonization, the point at which microbes start to settle into and onto the body. There is still scientific discussion about exactly how much microbial exposure happens before birth. Older thinking held that the womb was essentially sterile, and that colonization began only at birth. Newer research has raised more complicated possibilities, though this is still debated and difficult to study cleanly. What is clear is that birth itself is a major turning point. The moment a baby enters the world, the body is suddenly exposed to a huge range of microbes from the surrounding environment, from the mother, from skin, from air, from surfaces, and from feeding. That first wave of exposure matters. It helps shape which organisms arrive first, and first arrivals can influence what comes later. This is one of the oldest rules in ecology. Early settlers change the land for the ones that follow. In a forest after a fire, the first plants to return alter the soil, light and moisture in ways that affect later species. In the gut, early microbes can change acidity, consume oxygen, produce byproducts, and create conditions that make the environment more or less hospitable to others. The first settlers do not merely show up, they help build the stage. Mode of birth plays a role in this early microbial encounter. During vaginal delivery, a baby is exposed to microbes from the mother's birth canal and surrounding tissues. During caesarean delivery, the pattern of exposure is different, with more influence from skin and the immediate environment. This does not mean one child is doomed to microbial misery and the other instantly blessed with microscopic perfection. Biology is never that melodramatic, but it does mean that the early mix of microbes can differ, and researchers have observed differences in microbiome development associated with delivery method, especially in the early months. Over time, many influences continue shaping the microbiome, so birth is important, but it is not the only act in the play. Feeding is another major force. Breast milk is not simply food in the ordinary sense. It is an astonishing biological system, rich not only in nutrients but in immune factors, enzymes, and special carbohydrates called human milk oligosaccharides. These are particularly interesting because infants cannot digest them directly. Instead, they help feed certain beneficial microbes, especially species of bifidobacterium, which are often important early settlers in the infant gut. It is a wonderfully indirect strategy. The body does not just feed the baby, it also helps feed the bacteria that may support the baby. Formula-fed infants can also develop healthy microbiomes, of course, but the microbial patterns may differ because the nutritional landscape differs. Once again, the point is not moral judgment. The point is ecology. Feed different organisms differently, and you influence who thrives. This is worth sitting with for a moment because it reveals how deeply interconnected early life really is. The infant gut is not just being nourished, it is being introduced to partnership. The developing body is learning, through chemistry and contact, what kinds of microbial relationships it will carry into the months and years ahead. The gut lining is maturing. The immune system is learning what to tolerate and what to watch carefully. Microbial communities are shifting in response to milk, touch, household environments, siblings, pets, antibiotics, illness, and the general chaos of being a new human in a very populated world. At first, the infant microbiome is less diverse than the microbiome of an adult. That makes sense. A new settlement is not yet a great city. It has a few early arrivals, a limited menu, and a developing infrastructure. Over time, as the diet changes, especially when solid foods are introduced, the microbial world becomes more varied. New foods provide new substrates. Plant fibers begin to matter more. Different bacteria gain opportunities to grow. The immune system continues adjusting. By the time a child reaches the later early years of life, the microbiome has often become much more complex and more stable, though still not fixed forever. And that, perhaps, is one of the most important things to remember. The gut microbiome is built, but it is never entirely finished. It stabilizes, but it does not become untouchable. Throughout life, microbes continue arriving, leaving, rising, falling, and competing. Some remain long-term residents. Some pass through briefly. Some arrive with food and never truly settle. Some flourish only if the environment favours them. Your microbiome in adulthood is not identical to your microbiome in infancy, nor will it remain exactly the same throughout life. It reflects a long history of exposures and conditions. It is shaped by diet, medication, travel, age, illness, stress, infection, sleep, and all the other things that quietly alter the inner conditions of the gut. Now, when we say that bacteria come to live inside you, it may sound as if they are all moving into the same apartment building and decorating it in similar shades of beige. In reality, different bacteria prefer different niches. Some live closer to the mucus lining of the intestines. Others are more abundant in the intestinal contents themselves. Some like specific food sources. Some depend on the leftovers of others. Some are more common in oxygen-rich conditions, though the deeper colon tends to favour those that can handle little to no oxygen. This creates a layered community. Certain species help prepare the ground for others by breaking down compounds into smaller pieces. One bacterium's waste product may be another's dinner. The gut is full of these exchanges, tiny chains of use and reuse that make the whole system more efficient. This is why the idea of good bacteria is useful only up to a point. It is true that some bacteria are often associated with health-supporting functions. Bifidobacteria, for example, are often important in early life and linked with beneficial effects. Lactobacillus species, though not always permanent colonizers of the gut in large numbers, are familiar names in discussions of fermentation and probiotics. Other important groups in the adult gut include members of genera such as Faecalibacterium, Roseburia, Akkermansia, Bacteroides, and many others, each associated with particular metabolic roles or ecological patterns. But no single group tells the whole story. Health tends to correlate not just with one hero microbe, but with diversity, resilience, and functional balance. A rich ecosystem tends to cope better with stress than a fragile one. At the same time, there are bacteria that can become troublesome when conditions shift. Some are opportunists, harmless enough in low numbers or when the community is balanced, yet more dangerous when other microbes are wiped out or the immune system is strained. Clostridioides difficile is a famous example. It can cause serious disease, often after antibiotics have disrupted the normal microbiome and opened up ecological space. That does not mean antibiotics are bad in some sweeping theatrical sense. They save lives and remain essential medicine. But they can also disturb the microbial community, sometimes dramatically, because they do not always distinguish neatly between the target pathogen and the helpful residents nearby. It is rather like sending in a repair crew with very large boots. This brings us to one of the great themes of microbiome science: colonization resistance. A healthy microbial community can help prevent harmful organisms from taking hold. It does this partly by taking up space and consuming nutrients, and partly by shaping the environment through acids, antimicrobial compounds, and interactions with the immune system. In that sense, the first settlers and the later residents form something like a living defense network. They do not patrol with tiny flashlights, but they make life harder for unwelcome arrivals. When this system is intact, it can be remarkably effective. When it is disrupted, the gut may become more vulnerable. The immune system, meanwhile, is not standing apart from all this with crossed arms and a suspicious expression. It is actively learning from these microbial relationships. Early microbial exposure helps educate immune development. The body learns that not every foreign molecule is a threat. It learns tolerance where tolerance is appropriate, and vigilance where vigilance is needed. This matters because an immune system that reacts too strongly to everything would be exhausting and damaging. An immune system that reacts too weakly would be unsafe. Gut bacteria help shape that middle path. They provide signals, influence immune cells, and help reinforce the barrier between the intestinal contents and the tissues beyond. That barrier is another quiet marvel. The intestinal lining is only one cell thick in many places, which sounds alarmingly flimsy until you remember how much biological engineering goes into making it work. It is protected by mucus, joined by tight cellular connections, watched by immune cells, and supported by microbial products. Some bacteria help maintain this barrier by producing metabolites such as butyrate, a short-chain fatty acid that fuels certain colon cells and supports gut health. Others interact with the mucus layer in more direct ways. Akkermansia mucinifera, for example, is known for its relationship with mucus and has become something of a celebrity in microbiome research, which is not a phrase anyone would have expected to say a few decades ago. As the child grows, food broadens the microbial landscape even further. Solid foods bring starches, fibers, fats, proteins, and plant compounds into the system in new combinations. Some bacteria thrive on fiber and help ferment it into useful products. Others are more involved in protein breakdown. Some are encouraged by diverse plant intake. Others expand when the diet becomes narrow or heavily processed. This does not mean every meal instantly rearranges the whole microbiome like furniture during a domestic disagreement, but repeated patterns matter. The foods most often consumed help decide which microbes are regularly fed, and regularly fed microbes tend to stay in business. Household life also matters. People who live together often share aspects of their microbiomes. Close contact, shared environments, shared meals, and even shared pets can influence microbial exchange. A child growing up in a home with siblings, outdoor play, varied foods, and lots of ordinary exposure to the non-sterile world may encounter a different microbial landscape than a child in a more restricted environment. Again, this is not a matter of romanticizing dirt or suggesting that hygiene is some great modern mistake. The goal is not chaos. The point is that microbial exposure is part of life, and the body evolved in a world where total sterility was never the norm. There is also something wonderfully humbling in all this. We spend so much of modern life trying to control every variable, sanitize every surface, predict every outcome, and organize the body into neat categories. Then microbiology arrives and reminds us that health includes relationships with creatures too small to notice, influenced by birth, milk, food, touch, air, and the ordinary messiness of living among other beings. It is not a tidy story, but it is a rich one. By adulthood, the gut microbiome is usually relatively stable compared with infancy, But stable does not mean permanent. Travel can change diet and exposure. Antibiotics can reduce populations sharply. Illness can alter gut conditions. Aging can influence diversity and resilience. Stress can affect gut motility, secretion, appetite, sleep, and immune signaling, all of which ripple through the microbial environment. The first settlers are long gone in any literal sense, yet their role remains important because they helped shape the early trajectory of this inner ecosystem. They established patterns, influenced immune development, and participated in the long handoff from a newborn's emerging microbiome to an adult's more established one. And still, the story remains less like a static portrait and more like weather over a landscape. The terrain stays recognizable, but conditions shift. Some species rise in abundance, others fall. A new diet can encourage different fermenters. A course of antibiotics can temporarily flatten diversity. Recovery may be quick in some people and slower in others. Some microbes return because they were only reduced, not erased. Others may need help from diet and time to regain their footing. The microbiome has memory in a loose ecological sense. It tends to return toward familiar patterns, but not always completely and not always quickly. This is why people sometimes ask whether a microbiome can be fixed, as though it were a broken toaster. Usually the better way to think about it is support, not perfection. Healthy microbial communities are nurtured through conditions that favour balance and resilience. That often means diverse plant foods, adequate sleep, physical movement, careful use of antibiotics when needed, and an overall environment that does not constantly throw the gut into crisis. No one lives perfectly, and the microbiome does not require a saintly existence to function. It requires patterns that give beneficial organisms something to work with. If the beginning of this story feels intimate, that is because it is. The first settlers do not arrive in some abstract laboratory model. They arrive in a body just beginning life. They come through contact, through closeness, through feeding and breathing and being held. They help shape a system that will spend years learning what belongs, what nourishes, what harms, and how to maintain a peaceful and productive inner community. Long before most of us can form a memory, this silent settlement is already underway. And perhaps that is part of what makes the microbiome so compelling. It is not just a feature of digestion, it is a record of arrival. It is a living history of first contact between body and world, followed by years of negotiation between human life and microbial life. It begins in vulnerability and develops through relationship. It is affected by care, by circumstance, by biology, by luck, and by habit. In some ways, the gut microbiome is one of the earliest communities you ever belong to, even though you will never shake its hand or learn its names in the usual sense. Still, names do matter in science, and as we move further into this inner world, it becomes useful to understand not only how bacteria arrive, but what sorts of work they go on to do once they are there. Because settlement is only the beginning. Once the residents have moved in, the real business starts. Food must be processed, fibres must be broken down, useful compounds must be made from material the human body cannot handle alone. Some bacteria become the quiet labourers of digestion, turning stubborn plant matter into fuel and chemical signals the gut and body can actually use. And that is where this hidden world begins to grow even more interesting. Once those first settlers have arrived and the gut begins to feel less like an empty passage and more like a living place, the next part of the story becomes wonderfully practical. The residents need work, and some of the most important work in the whole microbial city belongs to the digesters. These are the bacteria that take the parts of food your own body cannot fully handle and do something useful with them. They break down fibres, ferment resistant starches, reshape leftovers from your meals, and in the process help feed not only themselves, but also you. It is one of the quietest and most important partnerships in the body. You eat the meal, the upper digestive tract does what it can, and then farther down, in the large intestine, the microbes take over where your own enzymes leave off. That handoff matters because human digestion, impressive though it is, has limits. The stomach can churn. The small intestine can break down fats, proteins, and simple carbohydrates with remarkable efficiency. Enzymes from the pancreas and the lining of the intestine can reduce many foods to absorbable pieces. But certain carbohydrates, especially many forms of dietary fibre, pass through this process largely undigested. That is not a failure. It is simply biology. Humans do not make all the enzymes needed to break apart the full range of complex plant compounds found in food. So when you eat beans, oats, lentils, onions, apples, barley, cooled potatoes, green bananas, or a broad range of vegetables, part of what you swallow is less a finished meal for you than a delivery of raw material for the bacteria waiting below. This changes the way it helps to think about fibre. Many people still imagine fibre as little more than bulk, something vaguely healthy that keeps the digestive system moving and gives breakfast cereal a moral advantage over pastries. There is some truth in that, of course. Fibre can help with stool bulk, bowel regularity, and the pace at which material moves through the gut. But that is only part of the story. Fibre is also food for microbes. It is one of the main energy sources for many of the bacteria that live in the colon, and when those bacteria digest it, they produce substances that can affect the health of the gut lining, the immune system, and metabolism throughout the body. This microbial digestion is usually called fermentation. In everyday life, the word fermentation may bring to mind bread, yogurt, sauerkraut, kimchi, or a jar at the back of the fridge that has become quietly alarming. In the gut, fermentation is the process by which bacteria break down carbohydrates in the absence of oxygen and generate smaller compounds as byproducts. Deep in the large intestine, oxygen is limited, which makes it a very suitable place for this kind of microbial work. The bacteria that thrive there are adapted to those low oxygen conditions and equipped with tools your own body does not possess. Those tools are enzymes, and in the microbial world, enzymes are everything. Bacteria that digest fibre carry genes for breaking apart tough, complex carbohydrates. Scientists often group many of these enzymes under the broad idea of carbohydrate-active enzymes, which is a very proper scientific phrase for what amounts to a collection of tiny molecular scissors, wedges, and pry bars. Different bacteria carry different sets of these tools. Some are very good at breaking down certain plant fibres, some prefer resistant starch, some work on pectins, some thrive on inulin, some start the job and others finish it. This creates a network of cooperation and dependency. A meal rich in varied plant foods is not handled by one heroic bacterium stepping forward in a neat uniform. It is handled by a whole community passing pieces along. Among the most important groups involved in this work are members of genera, such as Bacteroides, Ruminococcus, Prevotella, Roseburia, Eubacterium, and Faecalibacterium, along with many others. Each tends to have its own strengths. Bacteroid species are often regarded as excellent generalists. They are well known for their ability to break down a broad range of complex carbohydrates, especially those coming from plants. They possess large sets of genes devoted to carbohydrate breakdown and can adapt to different diets by shifting which systems they use. In some ways, they are like the versatile workers in the city, the crews that show up with a well-stocked toolbox and a calm expression that suggests they have seen this problem before. Ruminococcus species, particularly some members found in the human colon, are well suited to breaking down resistant fibres and certain plant cell wall materials. They are part of the reason tough plant matter does not simply drift through the body untouched. Prevotella species are often associated with diets rich in plant-based carbohydrates and fibres, though the exact pattern varies from person to person. In some individuals, especially those who regularly consume high-fibre diets, these bacteria may be more abundant and more actively involved in fermenting complex carbohydrates. What matters is not memorizing every microbial surname as if there will be an exam later tonight, but understanding that the gut hosts specialists, and those specialists respond to what they are regularly fed. Then there are the butyrate producers, a particularly interesting set of bacteria because of what they make from the work of fermentation. Butyrate is one of the short-chain fatty acids produced when fibre is fermented in the colon. Others include acetate and propionate. These molecules may be small, but their effects are not small at all. Butyrate, in particular, is an important fuel source for the cells lining the colon. Those cells, called colonocytes, rely heavily on it. When butyrate is available, it helps support the health of the intestinal lining, influences inflammation, and contributes to the integrity of the gut barrier. In simple terms, some of the bacteria in your gut take leftovers from your meal and convert them into fuel for the very tissue that houses them. It is a surprisingly tidy arrangement for something happening in the large intestine. Faecalibacterium prausnitzii is often mentioned here because it is one of the more studied butyrate-producing bacteria and is frequently associated with gut health. Rosebaria and Eubacterium species can also contribute to butyrate production. These bacteria are not digesting fibre merely to be helpful, of course. They are living their lives and extracting energy in the way evolution has shaped them to do. But the result is mutually useful. You provide fibre, they ferment it, you receive products that nourish your intestinal cells and influence physiology in helpful ways. It is a partnership built not on intention, but on shared advantage, which is often how the most durable biological relationships work. Acetate and propionate also matter. Acetate is the most abundant short-chain fatty acid in many people, and can circulate beyond the gut to be used in various tissues. Propionate travels largely to the liver and may influence glucose production and metabolic signaling. Together with butyrate, these compounds are part of the reason the digesters matter so much. They do not simply reduce fibre to waste. They transform it into molecules that the body can use, read, and respond to. That is why it is fair to say these bacteria help feed the body. They extend the digestive capacity of the human system and create energy-rich and signal-rich compounds from material that would otherwise be far less useful. This also helps explain why low-fibre diets can be such a problem for the microbiome over time. If the bacteria adapted to ferment fibre are not regularly fed, they may shrink in abundance. Other microbes that prefer different food sources may become more dominant. The overall production of beneficial short-chain fatty acids may fall. Diversity may decline. The community may become less resilient. None of this usually happens in a single dramatic evening after one disappointing dinner. Microbiomes respond to patterns, not to isolated moments. But a diet persistently low in diverse plant fibres can gradually starve the very bacteria that do some of the most useful work in the colon. By contrast, diets that include a wide range of plant foods tend to support a richer fermenting community. Different fibres feed different bacteria. Some microbes prefer beta-glucans from oats and barley. Some thrive on pectins from fruits. Some can make use of inulin and fructooligosaccharides found in foods such as onions, garlic, leeks, asparagus, and chicory. Resistant starch, found in legumes, whole grains, and cooked then cooled starches like potatoes or rice, can be especially valuable for certain fermenters. When you eat a variety of these foods, you're not just adding fibre in some abstract nutritional sense, you're broadening the menu for an ecosystem. And ecosystems tend to do better when the menu is not painfully repetitive. This variety matters for another reason as well. Microbial digestion often depends on cross-feeding, which is one of the lovelier concepts in gut ecology, once you get past the fact that it is happening in your intestines. One bacterium starts breaking down a complex carbohydrate into smaller fragments. Another bacterium then uses those fragments and produces different byproducts. A third may feed on those. The output of one becomes the input for another. This means the benefits of fibre are not always tied to a single species acting alone. They are tied to networks. A diverse microbiome can build complex metabolic chains, and those chains may be part of what makes the whole system robust. Imagine, for example, that one group of bacteria breaks a fibre down into lactate and acetate. Another group then uses those compounds to make butyrate. If the first group is present but the second is missing, the outcome changes. If both are present and well supported, the result can be quite different. The gut is full of such relationships. It is not unlike a small economy in which one workshop provides materials for the next. If several workshops close, the whole town feels it. The elegant part is that most of this happens without any involvement from your conscious mind. You do not need to supervise it. You're not expected to stand by with a clipboard while your bacteria negotiate the fate of a lentil. The speed of the large intestine also helps make this work possible. Compared with the small intestine, the colon moves contents more slowly. That slower pace gives bacteria time to act. Food residues sit long enough for fermentation to occur, for fibres to be broken down, and for short-chain fatty acids to be produced and absorbed. It is not glamorous, but it is efficient. Much of the energy and signalling value obtained from fibre depends on this microbial labour occurring in a part of the gut that gives the workers enough time to do the job properly. Of course, microbial fermentation does not produce only elegant molecules with reassuring names. It also produces gas. Hydrogen, methane, carbon dioxide and other gases can arise during fermentation, depending on the bacteria involved and the foods being processed. This is entirely normal, though perhaps less celebrated in polite conversation than butyrate. The more honest version of digestive health is that many beneficial processes come with side effects that are not especially romantic. A sudden increase in fibre intake can lead to bloating, discomfort, or more gas than usual precisely because the microbes are being given more material to ferment. In time, many people adapt as the community shifts, but the short-term result can feel as though the body is holding a small and argumentative meeting after dinner. That is one reason gradual change often works better than sudden dietary heroics. If someone who usually eats very little fibre suddenly decides to consume an enormous quantity of beans, bran and raw vegetables overnight, the microbiome may respond with enthusiasm but not elegance. The bacteria that can use these foods may begin fermenting more actively, but the system may need time to adjust. A slower increase gives the gut and its microbial residents a chance to adapt. The lesson, as with many things in biology, is that support tends to work better than shock. There is also an important difference between soluble and insoluble fibre, though the boundary is not always perfectly neat. Soluble fibres dissolve or swell in water and are often more fermentable. These can be especially useful for feeding bacteria. Insoluble fibres tend to add bulk and can influence stool form and transit. Both matter, but fermentable fibres are particularly important when discussing the digesters because they provide the raw material from which short-chain fatty acids and other metabolites are made. Resistant starch deserves special mention because it behaves less like ordinary digestible starch and more like a fermentable substrate that reaches the colon. It can be found in foods like legumes, oats, underripe bananas, and cooled cooked starches. Certain bacteria appear especially able to use it, and its fermentation may support beneficial metabolic effects. The effects of microbial digestion do not stop at the colon wall. Short-chain fatty acids can influence appetite signalling, blood sugar control, and inflammatory processes. They may interact with receptors on cells in the gut and beyond. They may affect the release of hormones involved in fullness and energy regulation. They may help calm excessive inflammation and support the maintenance of the gut barrier. This means the digesters are not merely handling waste disposal, they are producing molecules that participate in body-wide communication. A meal rich in fibre may eventually influence not just bowel habits, but the chemical conversation between gut, immune system, liver, and even the brain. This is where the phrase "feed the body" becomes especially accurate. Fibre itself may not be digested by your own enzymes, but its microbial breakdown products are meaningful to you. The bacteria are not digesting your food in some separate world that never touches the rest of the organism. They are contributing to a second stage of digestion, one that the human body relies upon for certain benefits. Without them, the energy and signalling value of many fibres would be largely inaccessible. The colon would receive material, but much less of it would be transformed into useful compounds. It is also worth noting that not every bacterium associated with carbohydrate digestion behaves in the same way across all contexts. Some species that help break down certain substrates may be beneficial in one microbial balance and less helpful in another. Some may produce metabolites that depend on what they are fed. Give a bacterium one type of substrate, and it may produce a generally beneficial pattern of compounds. Feed a very different diet and the outputs may shift. This is why microbiome science keeps returning to ecology rather than morality. The system is shaped by context, nutrients, relationships, and balance. It is not a simple scoreboard of heroes and villains. Even so, some broad patterns are reliable enough to hold on to. Diets rich in varied plant fibers tend to support fermentation and the growth of bacteria associated with short-chain fatty acid production. Diets low in fiber and high in heavily processed foods may support a less diverse community and reduce these fermentation benefits. A healthy colon often depends in part on being fed by microbial products, especially butyrate. And a resilient microbiome tends to require repeated, consistent nourishment from the kinds of carbohydrates that bacteria evolved to handle. There is another intriguing detail in all this. When fermentable fibre is scarce, some bacteria may turn more heavily toward the mucus layer lining the gut as a food source. That layer is a normal and important part of intestinal defence, and some microbes naturally interact with it, but a chronically poor diet may alter the balance in ways that are less favourable. This does not mean skipping vegetables for a week causes your colon to descend into some dramatic internal scandal. But over the long term, the health of the gut lining and the health of the microbial community are closely linked, and fibre helps protect that relationship by providing preferable food. Different populations around the world have provided striking examples of this. People eating traditional diets high in plant diversity and fibre often show microbiomes quite different from those eating highly processed low-fibre diets. Certain bacterial groups appear more abundant, and overall diversity may be greater. This does not mean the past was perfect, or that every traditional diet should be treated as a sacred microbial script. It does mean that the modern low-fibre pattern common in many industrialised settings is not necessarily what the gut microbiome is best adapted for. In some sense, many of our inner digesters are still waiting patiently for the menu they were designed to work with. Children, adults, and older individuals all depend on these bacterial processes, though the microbial community and its resilience may change over the lifespan. In early life, as solid foods are introduced, fermenting bacteria begin taking on larger roles. In adulthood, dietary patterns strongly shape which digesters remain abundant. In older age, changes in diet, medication use, gut motility, and general health can influence how well the fermenting community is maintained. The need for these bacteria does not vanish. If anything, the stability of their contribution becomes more important, because the gut lining, immune system, and metabolism all continue to rely on their work. The more one looks at this system, the less appropriate it seems to describe fibre as something passive, and the more appropriate it seems to describe it as a message and a meal. It is a message because it tells the microbiome what kind of environment this is going to be. It is a meal because it literally feeds organisms that, in turn, create useful compounds for the host. Every serving of legumes, whole grains, vegetables, fruits, nuts, and seeds is not just nutrition in the ordinary sense. It is also a vote for certain microbial workers, especially the ones that help turn rough plant structures into something the body can actually use. This, perhaps, is one of the most underappreciated forms of digestion in the whole body. The stomach receives a great deal of respect, though not always kindness. The small intestine does a great deal of absorbing, but the colon and its microbial residents perform a kind of rescue operation on what remains, salvaging value from complexity. They take what the upper gut could not finish and make it matter. They feed themselves, yes, but they also feed the tissue they live in and influence physiology far beyond their immediate neighbourhood. It is not too much to say that part of your nourishment depends on having the right microscopic workforce in place. And that workforce does not just appear in the same form in everyone. The abundance of fiber-digesting bacteria differs from person to person. Some people carry more of certain groups than others. Some microbiomes adapt quickly to dietary change, others shift more slowly, some produce more butyrate, others have different fermentation patterns. This variation helps explain why the same diet may not feel identical in every person. The food is the same, but the workers processing it are not. Your microbiome is part of the reason your body responds in its own way. Still, the general principle remains wonderfully steady. If you regularly feed the digesters, they tend to do their work more effectively. If you neglect them for long enough, the system becomes thinner, less productive, and often less resilient. It is an old ecological truth made personal inside the human body. Communities flourish when given resources. They weaken when starved. And because these particular communities live in your gut, their flourishing or weakening becomes part of your own biology. So while the body above may seem quiet tonight, there is likely a small and determined population in your colon sorting through the remains of your meals, clipping apart fibers, passing pieces along, trading metabolites, making short-chain fatty acids, and helping sustain the tissue that keeps the whole inner environment intact. They are the digesters, the workers who take complexity and turn it into usefulness. And useful as that is, digestion is still only one of their gifts, because some of the same bacterial worlds that help break down food also help manufacture nutrients the body needs, adding another layer to the quiet labour happening inside you. If the digesters are the quiet workers who take the rough leftovers of a meal and turn them into something useful, then the next group in this inner world feels almost like a set of hidden artisans. These are the vitamin makers, the bacteria that can help produce essential nutrients and contribute in ways both direct and indirect to the body's long and careful work of staying alive. It is one of the more surprising parts of the gut story. Most people think of vitamins as something that arrives from outside, from fruits, vegetables, grains, meats, eggs, supplements, or the uneasy promise printed on the side of a cereal box. And that is true. We depend heavily on diet for vitamins. But once food enters the body, the story does not end with a bite. Deep in the gut, microbes are also at work, making certain vitamins, reshaping nutrients, and influencing how much value the body can draw from what passes through it. This does not mean your bacteria are running a complete nutritional factory, capable of replacing food altogether. Sadly, no microscopic workforce is going to excuse a person from eating a decent meal. Biology is clever, but it has not yet arranged things so that one can live indefinitely on biscuits and optimism, while the colon heroically fills in the missing vitamins. The truth is more subtle and more interesting. Gut microbes help with nutrient production, and in some cases they may make a meaningful contribution to the body's vitamin economy. In other cases, they produce vitamins that are important within the microbial community itself, or that may only partly benefit the host depending on where in the gut they are made and how absorption works. It is not a fairy tale of perfect microbial generosity. It is a layered partnership full of limits, opportunities, and very practical chemistry. To understand why this matters, it helps to remember what vitamins are. Vitamins are organic compounds the body needs in small amounts for essential functions. They help enzymes work, support metabolism, assist in building and repairing tissues, aid the nervous system, participate in blood clotting, help make red blood cells, and do countless other jobs so ordinary and so constant that we only notice them when something begins to fail. Unlike protein, fat, or carbohydrate, vitamins are not major sources of energy. They are more like the tiny tools and chemical keys that let the machinery run. Without them, the body's processes become slower, less reliable, and eventually damaged. Some vitamins must come almost entirely from the diet because the human body cannot make enough of them on its own. Others can be produced in small amounts or transformed from precursors. And then there is the microbial contribution, especially in the large intestine, where bacteria can synthesize several vitamins, particularly vitamin K and a number of B vitamins. The exact mix depends on which bacteria are present what they are being fed, what the conditions of the gut are like, and how the larger ecosystem is functioning. Different microbes have different abilities. Some can make folate, some can make biotin, some can produce riboflavin, niacin, pantothenic acid, pyridoxine, or forms of vitamin K. Some may even make vitamin B12, though that comes with a complicated catch involving where absorption takes place in the human gut. That catch is important because it keeps the story honest. The gut microbiome does make vitamins, but the value of those vitamins to the human host depends on location and timing. Many vitamins are absorbed primarily in the small intestine, especially in the upper and middle regions, before food reaches the colon where the densest bacterial populations live. So if a bacterium in the large intestine produces a vitamin, that vitamin may not always be efficiently absorbed in the same way as one consumed earlier in the digestive process. This means microbial production is not always equivalent to dietary intake. Some vitamins made in the colon may still matter, some may be absorbed to a degree, some may help local tissues or other microbes, some may not contribute enough to fully meet human needs. The microbiome supports nutrition, but it does not abolish the need for food. Even so, the contribution can be meaningful, and perhaps the most famous example is vitamin K. Vitamin K is actually a group of related compounds involved especially in blood clotting and bone metabolism. One form, often called vitamin K1, comes mainly from leafy green plants. Other forms, especially certain menachinones often called vitamin K2, can be produced by bacteria. Gut microbes are among the organisms capable of making these compounds, and for a long time this has been recognized as one of the clearest examples of microbial vitamin production in the human body. The exact extent to which bacterial vitamin K contributes to overall status can vary, and dietary sources still matter greatly, but it remains a striking reminder that the gut is not merely taking nutrients apart, it is also helping create some. Then there are the B vitamins, an entire family of compounds involved in metabolism, nervous system function, blood cell production, DNA synthesis, and more. Here the microbiome becomes especially interesting. Many gut bacteria carry the genes needed to synthesize one or more B vitamins. Folate, also known as vitamin B9, is one of the better-known examples. Folate is vital for DNA synthesis and cell division, which is why it is especially important in periods of growth and pregnancy. Certain bacteria can produce folate in the gut, and researchers have long been interested in how much this may support the host under normal conditions. The answer is not always simple, but the capacity is there, and it is part of the broader microbial contribution to nutrient metabolism. Biotin, or vitamin B7, is another example. Biotin plays a role in fat metabolism, carbohydrate metabolism, and several enzyme systems. Gut microbes can produce it, and some species are particularly good at doing so. The body also gets biotin from foods such as eggs, nuts, seeds, and legumes. But microbial production adds another layer to the picture. Riboflavin, or vitamin B2, and niacin, or vitamin B3, can also be made by certain bacteria. Pantothenic acid, vitamin B5, is involved in coenzyme A and energy metabolism, and again some microbes can synthesize it. Pyridoxine and related forms of vitamin B6 are also part of this microbial toolbox. In other words, the gut hosts a small but chemically impressive guild of nutrient makers, each contributing to a broader pattern of metabolism. Vitamin B12, however, is where the story becomes especially good at ruining simplistic headlines. Certain bacteria can indeed synthesize B12 or B12-like compounds. The difficulty is that in humans, vitamin B12 is mainly absorbed in the terminal ileum, which is the last part of the small intestine, with the help of intrinsic factor produced by the stomach. Most of the dense bacterial production occurs later in the large intestine. So although bacteria can make B12, much of that production may happen too far downstream to be efficiently absorbed by the host in the ordinary way. It may benefit the microbial community, and it may still matter in indirect ways, but it is not wise to imagine the colon cheerfully covering all your B12 needs while you ignore the rest of nutrition. This is one of those cases where microbiology offers something fascinating, but also insists on anatomical realism. The relationship between microbes and vitamins goes beyond simple production. Gut bacteria also help release nutrients from food, transform compounds into more useful forms, and influence the overall environment in which digestion and absorption take place. Some bacteria can free certain plant-bound compounds that would otherwise be harder for the body to access. Others affect acidity, enzyme activity, or the condition of the gut lining, which in turn can influence nutrient handling. A healthy gut barrier and a well-regulated intestinal environment are not just good for comfort. They are part of the conditions that make nutrition work properly. Vitamins do not float through the body by magic. They rely on intact tissues, appropriate transport systems, and a digestive tract that is functioning reasonably well. The microbiome helps shape those conditions. This is one reason the idea of vitamin makers cannot be separated from the rest of gut health. A microbiome that is diverse, balanced, and well-fed is more likely to support efficient nutrient metabolism than one that is disturbed, depleted, or inflamed. Repeated antibiotic use, severe illness, poor diet, chronic gut inflammation, or prolonged stress can all affect the microbial community. When the community changes, the range of metabolic activities in the gut can change with it. Some vitamin-producing organisms may decline, some pathways may become less active. The environment in which nutrients are absorbed may become less favourable. None of this means one rough month instantly produces some dramatic nutritional collapse, but over time these relationships matter. The body's nutrient status depends on what enters the mouth, what gets digested, what gets absorbed, what gets used, and what the microbes in the gut are doing along the way. It is also worth noticing that microbial communities have their own nutritional lives. Bacteria need vitamins too. Some can make certain vitamins themselves, some depend on other microbes to supply them, some compete for the same compounds. This means that vitamin metabolism in the gut is not always a simple gift from bacteria to host. It is also part of the internal economy of the microbiome. One species may produce a vitamin that another species needs. A third may scavenge that product. A fourth may be able to make it under some dietary conditions but not others. So when we speak of vitamin makers, we're also speaking about community structure, about trade and dependency within the microbial city. The human host benefits from this chemistry, but the microbes are not organising the whole system with human nutrition as their sole and noble purpose. They are surviving, and their survival often becomes part of our advantage. This is why diet still matters so much even in the presence of microbial help. The microbes can make certain nutrients, yes, but they also depend on being fed. A fibre-rich, varied diet tends to support a more diverse and metabolically capable microbiome. Plant foods provide not only vitamins directly, but also the fermentable fibres and complex carbohydrates that help beneficial bacteria thrive. Whole grains, legumes, vegetables, fruits, nuts and seeds help create the conditions under which many helpful bacterial functions can continue, including the production of vitamins and short-chain fatty acids. In a sense, the better you feed the ecosystem, the more capable that ecosystem becomes. A narrow, low-fibre, heavily processed diet may not only supply fewer vitamins directly, it may also reduce the microbial support systems that help optimize nutrient handling. There is something elegantly circular about this. You eat foods rich in plant compounds and nutrients. Those foods help nourish microbes. Those microbes then contribute to the production of other useful compounds, support the gut lining, and help maintain the conditions under which nutrients are handled well. Health feeds health. Or, to put it in less polished but perhaps more memorable terms, your vegetables are not just helping you directly. They are also helping your tiny internal workforce keep the place running. Some fermented foods have drawn attention here as well. Yogurt, kefir, sauerkraut, kimchi, tempeh, miso, and other fermented foods may introduce live microbes or beneficial microbial products depending on the food and how it is prepared. These foods can also contain vitamins produced during fermentation itself. Certain fermented foods may supply vitamin K2 or some B vitamins more readily, because the microbes involved in fermentation have already done part of the work outside the body. This does not mean every jar of something pleasantly sour is a miracle cure sent by the gods of digestion, but fermented foods can be a valuable part of the larger nutritional picture. They remind us that microbes have always helped humans with food, both outside and inside the body. There are also times when the relationship between vitamins and bacteria becomes medically very obvious. Broad-spectrum antibiotics, for example, can disrupt the gut microbiome. In some situations, this disruption can affect vitamin K status, particularly if dietary intake is poor or other risks are present. This is one reason vitamin K has historically been discussed in the context of antibiotic use, especially when bleeding risk is a concern. The body is usually quite resilient, and one ordinary course of antibiotics does not mean the nutritional foundations of civilization have crumbled. But it does reveal that microbial contributions are not imaginary. Remove the microbes, and some consequences become easier to see. The same broader lesson appears in conditions that affect the gut lining itself. If the intestinal surface is inflamed, damaged, or functioning poorly, nutrient absorption can suffer even when the diet looks acceptable on paper. The microbiome, immune system, and gut barrier are closely linked, so bacterial imbalance can ripple into nutrition not only through reduced vitamin production, but through impaired handling of nutrients more generally. Deficiency is rarely caused by one thing alone. It may involve intake, absorption, inflammation, medication, disease, and microbial change all at once. The body is less like a row of independent departments and more like a company in which everyone keeps borrowing each other's staplers and occasionally causing system-wide confusion. It is also useful to say plainly that microbial vitamin production is not a reason to assume all supplements are unnecessary, or all deficiencies can be solved by fixing the gut. Some deficiencies are serious and require direct treatment. Iron deficiency, B12 deficiency, folate deficiency, and others can cause major problems and may need targeted medical care. The microbiome is part of the picture, but it is not the entire picture. That balance matters because microbiome discussions sometimes drift toward magical thinking, and the gut, interesting as it is, does not deserve to be turned into a mythology. It deserves something better than that. It deserves accurate wonder. Accurate wonder is available in abundance here. Consider the sheer oddness of it. Inside the colon are microbes using genetic instructions evolved over vast spans of time to build molecules your own body needs in order to run enzymes, divide cells, clot blood, and maintain nervous system function. They do this without your awareness, without ceremony, and generally without waiting for thanks. They are part of a distributed chemistry lab built into the human digestive tract. Not a complete lab, not a perfect one, and not one that removes the need for good food, but a real and consequential one all the same. Some researchers have even explored whether targeted probiotic strains might one day be used to enhance the production of certain vitamins in the gut. There is active interest in bacteria that naturally produce folate or riboflavin, for example, and whether they might be used in foods or therapies. This is a promising area, though one that still requires careful study. As always in microbiome science, the temptation is to rush from an intriguing mechanism to a grand promise. The wiser approach is patience. The biology is rich enough without overstatement. It is enough to know that the microbiome can contribute to nutrient production, and that, under the right conditions, this contribution may matter in meaningful ways. And those right conditions usually begin in wonderfully ordinary places. They begin with meals that contain real variety. They begin with fibre, because many vitamin-supporting microbes do better in communities built on fermentation and plant diversity. They begin with not treating the gut as an afterthought. They begin with recognizing that food does not merely feed human cells directly. It also feeds an ecosystem whose chemistry becomes part of your chemistry. There is no need to turn dinner into a moral trial or a spiritual quest. It is enough to understand that repeated patterns shape repeated outcomes, and that the microbes making vitamins tend to prefer a diet that looks a great deal like the diet nutrition science has been recommending for years anyway. Older age offers another layer to the story. As people age, dietary patterns may narrow, appetite may change, medication use often increases, and the microbiome can become less diverse in some cases. This may influence vitamin status indirectly through both intake and microbial function. Supporting the microbiome in later life may therefore matter not only for digestion and immune balance, but for nutrient handling as well. Again, nothing here suggests that bacteria alone can solve the challenges of aging, but they remain part of the larger network that helps the body remain nourished. In all this, there is a gentle lesson about how life works. The body is not a self-contained machine that handles everything alone. It is helped constantly by relationships, by exchanges, by organisms too small to see, by layers of cooperation built so deeply into ordinary function that we forget they are there. Vitamins feel like such small things until they are missing. Microbes feel like such small beings until you begin to notice how much they influence. Put the two together and you get one of the loveliest quiet truths in biology. Some of what sustains you is being made in darkness by invisible residents from the remains of meals you have already forgotten eating. And as that nutrient-making work goes on, another responsibility is unfolding alongside it. Because if the digesters and vitamin makers help feed and support the body, there must also be organisms helping guard the territory, holding the line against less welcome arrivals, using crowding, chemistry and cooperation to make the gut a harder place for harmful intruders to take over. If the gut were only a place of digestion, it would already be impressive enough. But it is not only a place where food is taken apart and useful compounds are made. It is also a borderland, a place where the body meets the outside world in one of the most direct and vulnerable ways imaginable. Every time you eat, drink, swallow a pill, or absent-mindedly put something in your mouth that absolutely did not belong there, the digestive tract becomes an entry point. Most of what passes through is harmless, much of it is useful, some of it is helpful, and some of it, unfortunately, would be quite happy to make a mess of things if given the chance. That is why the gut needs protectors, not in the dramatic sense of tiny microbes marching around in polished armour with banners flying, though that would certainly improve the reputation of microbiology. What it needs are resident bacteria that make life difficult for harmful invaders, help maintain order, support the barrier of the intestine, and work with the immune system to keep the whole place from turning into chaos. This protective role is one of the most important gifts of a healthy microbiome. It is also one of the easiest to overlook because when it works well, nothing obvious happens. You do not feel the absence of infection as a daily event. You do not wake up and think, "What a lovely morning, my colonization resistance appears to be functioning beautifully." Most of us go about our day with no idea that one of the reasons harmful microbes fail to take hold is because the residents already living in the gut are doing what established communities so often do. They are occupying space, using up resources, shaping the environment, and making it harder for strangers to move in and start trouble. This is the broad idea behind what scientists call colonization resistance. It sounds a little formal, perhaps the kind of phrase one might expect from a stern microbiologist wearing sensible shoes. But the idea itself is quite simple. A healthy microbial community helps resist the growth of harmful organisms. It does this through several overlapping strategies. Some bacteria compete for nutrients so effectively that invaders struggle to find enough fuel. Some cling to surfaces and occupy space that would otherwise be available for pathogens. Some produce acids or antimicrobial compounds that make the environment less welcoming to unwanted microbes. Some help maintain the mucus layer and intestinal barrier. Some interact with the immune system in ways that strengthen the body's response to real threats, without causing constant, unnecessary inflammation. Protection in the gut is not one single act. It is a whole pattern of crowding, chemistry, maintenance, and education. To appreciate why this matters, it helps to remember what the gut is up against. The digestive tract is not sealed off like a vault. It is more like a controlled gateway with an enormous amount of traffic. Food carries microbes. Water can carry microbes. Hands, surfaces, saliva, and countless ordinary interactions with the world bring microbial exposure all the time. Most of this is harmless, and some of it is part of normal life. But the gut must remain selective. It cannot simply allow anything that arrives to settle in and flourish. If it did, every questionable sandwich and every travel mishap would become the beginning of an internal disaster. The body has its own defences, of course. Stomach acid kills many organisms before they ever reach the intestines. Digestive enzymes and bile create difficult conditions for many microbes. The intestinal lining is covered by mucus. Immune cells stand watch. Antibodies are secreted into the gut. Tight junctions between cells help prevent the contents of the intestine from drifting casually into the bloodstream as if the whole arrangement were a social suggestion rather than a border. All of these are important, but the resident bacteria form another layer of defense, one that is living, adaptive, and always present when the system is healthy. One of the simplest protective strategies is competition for nutrients. Harmful bacteria, like all living things, need food. If the gut is already populated by dense, well-adapted microbial communities, much of the available fuel is already being used. This leaves fewer opportunities for invaders. A pathogen entering the gut is not stepping into an empty dining hall with a reserved table and a warm welcome. It is arriving in a crowded marketplace where the best supplies are already spoken for. If the resident microbes are abundant and active, harmful organisms may find it difficult to gain enough energy to multiply effectively. Competition for space is just as important. Many bacteria attach to the mucus layer or live close to the intestinal lining. If protective or harmless resident species are already occupying these niches, harmful microbes have fewer places to establish themselves. This matters because successful colonization often depends on attachment. A microbe drifting through the gut may cause less trouble than one that can latch onto the intestinal surface, resist being swept along, and begin multiplying in place. Resident bacteria, by simply being there first and staying there consistently, help reduce the number of available footholds. Then there is chemistry, which is where the protectors become especially interesting. Many beneficial bacteria produce compounds that alter the local environment. Lactic acid bacteria, for example, can help lower pH in certain settings by producing lactic acid. A more acidic environment can be less favourable for some harmful organisms. Other bacteria produce short-chain fatty acids such as acetate, propionate, and butyrate during fermentation. These molecules do many useful things, including supporting the gut lining and influencing inflammation, but they can also contribute to an environment that is less comfortable for certain pathogens. The protectors are not merely taking up room, they're changing the rules of the neighbourhood. Some resident bacteria also produce more direct antimicrobial substances, often called bacteriocins. These are compounds made by bacteria that can inhibit or kill other bacteria, usually closely related ones or competitors in the same environment. It is a wonderfully petty and efficient system. Imagine a crowded apartment building in which several tenants have quietly learned how to make cleaning products that only their least favourite neighbours cannot tolerate. The gut contains many such small acts of microbial rivalry. And although bacteriocins are not the only way protection happens, they are part of the chemical landscape that can make life harder for invaders. Lactobacillus species are often discussed here, especially because many are known from fermented foods and probiotic products. They are not always permanent major residents of the colon in every person, but they can be important in certain parts of the gut and in specific contexts. They produce lactic acid, can influence pH, and in some cases produce antimicrobial compounds. Bifidobacterium species are another familiar group, especially important early in life but also valuable throughout it. They help ferment certain carbohydrates, produce helpful metabolites, and are often associated with gut environments that are less favourable to pathogens. Again, the point is not that one genus deserves all the applause while the rest of the microbiome waits backstage. It is that several groups of bacteria contribute to the protective function in overlapping ways. Bacteroid species, which are often central players in the adult gut, can also contribute to defence through competition and ecological stability. Some members of this group help occupy niches so effectively that harmful organisms struggle to break in. Other important groups include certain Clostridia, not the harmful sort people worry about in food poisoning headlines, but beneficial members that support immune balance and barrier health. Faecalibacterium prausnitzii, often mentioned for its association with butyrate production and lower inflammation, may also support protective conditions indirectly by helping maintain the gut environment in a calmer and healthier state. In microbiology, protection often comes not from aggression alone, but from stability. That stability matters enormously for the intestinal barrier. The gut lining is thin, busy, and under constant pressure to do two things at once. It must allow nutrients to pass through while also keeping harmful microbes, toxins, and undigested material from crossing where they do not belong. This balancing act depends on healthy intestinal cells, tight junctions between them, a well-maintained mucus layer, and proper communication with immune cells. Beneficial bacteria support all of this. Some stimulate mucus production, some help feed colon cells by producing butyrate, some influence genes involved in barrier function, some reduce unnecessary inflammation that might otherwise damage the tissue. The protectors are not just bouncers at the door, they are also part of the repair crew, the maintenance staff, and the people making sure the hinges do not come off. The mucus layer deserves a little more attention because it is one of the most elegant pieces of this whole system. It covers the intestinal lining like a soft protective coat. In parts of the colon, it forms a crucial separation between the dense bacterial population in the gut contents and the epithelial cells beneath. Beneficial bacteria help maintain the conditions that preserve this layer. Some interact with it closely, some feed on its components in balanced ways, and some support the cells that produce it. When the microbiome is healthy, the mucus layer is part of a working relationship. When the system is disturbed, this protection can weaken. Pathogens may then find it easier to approach the lining, attach, and provoke trouble. This is one reason a disrupted microbiome can make a person more vulnerable to infection. Antibiotics are the clearest and most dramatic example. They're invaluable medicines, often life-saving, and there is no sense in pretending otherwise. But they can also reduce populations of beneficial resident bacteria, sometimes quite sharply. When that happens, colonization resistance weakens. Nutrients become more available, space opens up, antimicrobial compounds are reduced. The barrier may be less well supported. In those moments, opportunistic pathogens can take advantage. Clostridioides difficile is one of the most famous examples. It can exist quietly in some settings, but after antibiotics disrupt the normal microbiome, it may grow rapidly and produce toxins that cause severe diarrhea and inflammation. The problem is not that antibiotics are evil. The problem is that when the local population is knocked back, someone unpleasant may notice the vacancy. This tells us something profound about health. Sometimes protection is not about adding a new defense. It is about preserving the defense that was already there. The healthy microbiome is not a decorative feature of digestion. It is part of what keeps the gut from becoming easy territory for harmful organisms. When we speak of good bacteria, that is often what we mean. Not morally good, certainly not pure in some storybook way, but beneficial to the host because their presence helps keep the system resistant, balanced, and difficult to invade. The immune system is deeply involved in this relationship. Beneficial bacteria do not simply block pathogens by crowding them out. They also help teach the immune system how to respond appropriately. The gut contains a large portion of the body's immune activity, and this makes sense when you consider how much foreign material passes through it. The immune system must be alert without being hysterical. It must tolerate food and helpful microbes while remaining ready to respond to dangerous ones. Beneficial bacteria help shape this balance. They influence immune cells, help promote regulatory responses, and support the production of secretory IgA, an antibody that can bind microbes and help control their behavior at the mucosal surface. This immune education begins early in life and continues through adulthood. A healthy microbial community helps train the body not to launch unnecessary attacks on every harmless stimulus, while still supporting vigilance against true threats. If this training is disturbed, immune responses may become less well regulated, inflammation may increase, barrier function may weaken, conditions become more favourable to pathogens not only because the bacteria are gone, but because the immune and tissue systems they help support are less steady. Protection in the gut is therefore a shared project. The bacteria and the immune system are in conversation all the time. That conversation becomes especially important when pathogens do arrive. Harmful microbes such as Salmonella, certain pathogenic strains of Escherichia coli, Shigella, Campylobacter and others have developed strategies to invade, attach, evade defences or produce toxins. They are not easily discouraged under all conditions, but a healthy microbiome can make their job much harder. A dense community of resident bacteria may consume the nutrients these pathogens need. The environment may be more acidic or chemically hostile. The mucus layer may be strong. Immune readiness may be better tuned. The pathogen still has its tricks, but the playing field is much less favourable. There is also a less dramatic but equally important protective role against overgrowth by organisms that are not always classic pathogens. Some microbes are opportunists. In small numbers or under normal conditions, they may do little harm. But if the ecosystem shifts, they can expand in ways that become troublesome. Certain Enterobacteriaceae, for example, may bloom when inflammation rises or the normal microbial balance is disturbed. Some yeasts can also overgrow in particular circumstances. The protectors help prevent these population swings by maintaining a stable and competitive environment. In ecology, resilience often matters as much as force. A stable system resists takeover not because it is violent, but because it is full, organised, and functioning. Diet strongly affects this protective capacity. A fibre-rich, varied diet tends to support beneficial bacteria that ferment plant material and produce short-chain fatty acids, which in turn support barrier health and balanced inflammation. A diet low in fibre and high in heavily processed foods may reduce these benefits and favour conditions that are less protective. This is not because one missed salad causes immediate surrender at the intestinal gates. The gut is more resilient than that. But repeated dietary patterns matter. Feed the protectors, and they tend to remain numerous and effective. Starve them for long enough, and the neighbourhood changes. Stress, sleep, illness, and medications matter too. Chronic stress can affect gut movement, secretion, appetite, and immune signalling. Poor sleep can influence inflammation and metabolism. Illness may alter the gut environment directly. Some medications, including proton pump inhibitors and antibiotics, can shift the microbial community or the chemical conditions of the digestive tract. All of these factors affect how strong colonization resistance remains. Protection in the gut is not a static fortress wall. It is a living system that responds to the broader life of the body. This is why some of the most effective support for gut defense looks almost ordinary. Diverse plant foods, adequate sleep, reasonable use of antibiotics when needed, rather than casual use when they are not. Movement, recovery after illness. In some cases, fermented foods or targeted probiotics may help support certain functions, though the results vary and are often more modest than the labels would have one believe. The grander claims of modern wellness culture often suggest that the gut is one dramatic smoothie away from spiritual renewal. Real biology is less theatrical and more dependable. Support the ecosystem consistently, and it tends to do what ecosystems do. It stabilizes, it resists, it recovers better. It is also important not to confuse the phrase good bacteria with perfection. Even beneficial resident microbes can become problematic if they end up in the wrong part of the body. A harmless gut commensal entering the bloodstream or urinary tract is no longer simply minding its own business. Context matters. The protectors are beneficial where they belong, and in the relationships they normally maintain. The body depends on order as much as on the organisms themselves. A well-kept gut ecosystem is useful precisely because it is in the gut, in balance, doing the jobs that evolved there. There is something reassuring in realizing that defense does not always require dramatic force. Much of the protection in the body comes from ordinary steadiness. The resident bacteria do not wait passively until disaster strikes. Their very presence is part of the defense. They are there first, they eat first, they attach first, they shape the chemistry before an invader arrives. They help maintain the border before it is tested. They support the immune system before it is challenged. That is often how the best protection works. It makes trouble less likely long before trouble has a chance to announce itself. And that may be one of the more elegant lessons hidden inside the gut. Health is not only about fighting off enemies after they appear, it is also about maintaining conditions in which enemies find it difficult to thrive at all. A healthy microbiome does not guarantee perfect protection, of course. Foodborne infections still happen. Travel still introduces surprises. Bodies still get sick. But the difference between an empty, disturbed ecosystem and a full, well-functioning one can be enormous. One is an opportunity, the other is an obstacle. So tonight, while the body seems quiet from the outside, there is likely a steady, invisible order being kept within. Bacteria are occupying space, using nutrients, supporting mucus, producing acids, shaping immune messages, and making the intestinal world harder for harmful organisms to conquer. They are the protectors, not because they are noble in the human sense, but because their ordinary life inside you creates safety. And that safety is not only about blocking invaders, it is also about teaching the body how to recognize friend from threat, calm from danger, and signal from noise, which leads naturally to the next part of the story, because some of these same microbes do not merely defend the gut, they help train the immune system itself. If the protectors help keep harmful invaders from gaining ground, then the next layer of the story is even more intimate because some of the bacteria living in the gut do not simply defend the border. They help educate the very system that decides what a threat is in the first place. This is one of the most remarkable parts of the relationship between the microbiome and the human body. The immune system is often imagined as a kind of internal army, always alert, always prepared, always scanning for danger, and there is some truth in that image. But an immune system cannot survive on vigilance alone. It also needs judgment. It has to know when to react strongly, when to remain calm, when to tolerate harmless substances, and when to stand down so that daily life inside the body does not turn into a permanent state of alarm. Gut bacteria help shape that judgment. They are not merely passengers benefiting from the shelter of the intestine. They are among the trainers of one of the body's most important and most delicate systems. This matters because the gut is one of the busiest meeting places between the body and the outside world. Food arrives, water arrives, microbes arrive. Fragments of plant compounds, proteins, fats, sugars, and countless foreign molecules pass through the intestinal tract every day. The immune system cannot treat every one of these encounters as a full emergency. If it did, eating breakfast would feel like a siege. The body would be locked in chronic inflammation, tissues would suffer constant damage, and ordinary life would become chemically chaotic. At the same time, the immune system cannot be too relaxed. Real pathogens do enter through the gut, and when they do, the body must respond effectively. So the task is not simply to attack. The task is to discriminate. The immune system must learn the difference between danger and harmlessness, between invasion and routine exposure, between a true threat and a spoonful of lentils doing their best. That learning begins early. The immune system is not born fully informed about the world. It develops through exposure, signaling, and repeated encounters with the body's own tissues, harmless substances, and microorganisms. The gut microbiome plays an enormous role in that education. From the earliest months of life, microbes begin interacting with immune cells, influencing how those cells mature and what kinds of responses become more or less likely. The developing immune system is, in a sense, being introduced to its neighbours. It is learning that not everything foreign is hostile and that peace, when appropriate, is not weakness but intelligence. This process is especially important in the gut because a large portion of the immune system is concentrated there. The intestinal lining is surrounded by immune tissues and immune cells that continuously sample the environment. There are specialised regions such as Peyer's patches in the small intestine, collections of lymphoid tissue that help monitor what is passing through the gut. There are dendritic cells that extend processes to sample microbial material. There are macrophages, T cells, B cells, and other immune participants moving through this landscape, responding not only to pathogens but also to the steady presence of resident bacteria and the molecules they produce. The immune system in the gut is not waiting in silence until disaster arrives. It is involved in constant low-level conversation. Gut bacteria are part of that conversation in several ways. They interact directly with immune cells through molecules on their surfaces. They release metabolites that affect immune behaviour. They help shape the mucus layer and the intestinal barrier, which in turn influences how the immune system interprets what it encounters. They also compete with pathogens and reduce inflammation-driving disturbances, which gives the immune system a steadier environment in which to function. In short, the trainers do not merely stand in front of a classroom with a pointer and a chalkboard. They shape the conditions under which the immune system learns. One of the most important lessons gut bacteria help teach is tolerance. In immunology, tolerance does not mean indifference or laziness. It means the ability to recognize something as harmless and avoid mounting a destructive response against it. This is essential in the gut. The body must tolerate food proteins, ordinary dietary compounds, and the resident microbiota themselves. If it reacted to all of these with full inflammatory force, the result would be tissue injury and ongoing dysfunction. Beneficial bacteria help promote the development of regulatory immune responses, especially through certain types of T cells known as regulatory T cells or TREGs. These cells help suppress excessive immune activation and maintain balance. Several bacterial groups appear especially important in encouraging these calming responses. Certain members of the Clostridia class, particularly clusters that are common in the healthy colon, have been shown in studies to support the development of regulatory T cells in the intestine. These bacteria help create an environment in which immune overreaction becomes less likely. Bacteroides fragilis, a well-known resident of the gut, has also drawn attention because of a molecule called polysaccharide A, which appears to influence immune development and support balanced immune responses. Here again, the microbiome reveals itself not as a passive population but as a chemical teacher, presenting signals that help instruct immune cells in how to behave. Short-chain fatty acids play a major role in this training as well. These are the same fermentation products made when bacteria break down dietary fibre, particularly butyrate, acetate, and propionate. They are often discussed in the context of digestion and barrier support, but they also matter for immune function. Butyrate, in particular, has anti-inflammatory effects and can influence the behaviour of immune cells in ways that promote tolerance and reduce unnecessary activation. It helps support regulatory T cells, affects gene expression, and contributes to the maintenance of a calmer immune environment in the gut. So when fibre-feeding bacteria ferment plant material and produce butyrate, they are not only feeding colon cells, they are also helping shape the tone of the immune system. This creates one of the clearest examples of how diet, microbes, and immunity are linked. A varied fibre-rich diet supports bacteria that ferment fibre into short-chain fatty acids. Those fatty acids then help support regulatory immune responses and barrier health. A diet low in fibre may reduce those benefits and shift the immune environment in less favourable directions. It is not that one bowl of oats turns a reckless immune system into a wise elder overnight, but over time, repeated patterns of diet help influence the microbial community, and that community helps influence the immune system. The training is ongoing. Another vital lesson the microbiome teaches is restraint in the face of harmless microbial presence. The gut contains trillions of bacteria, most of them harmless or helpful, and the immune system has to live among them without launching total war. This is no small task. It requires a kind of armed peace, a balance in which the immune system remains alert yet controlled. Beneficial microbes help reinforce this balance by supporting the mucus barrier and stimulating the production of secretory immunoglobulin A, or IgA. This antibody is released into the gut and combines microbes and microbial products, helping keep them contained within the lumen and mucus layer without creating the kind of inflammatory storm that would damage tissue. IgA is one of the great quiet diplomats of the immune world. It interacts with microbes, helps shape which ones persist, and maintains order without always escalating to violence. The mucus layer itself is part of the teaching environment. It separates most bacteria from the epithelial cells lining the gut, reducing constant direct contact, while still allowing immune sampling and communication. Beneficial bacteria support this layer in several ways, including stimulating mucus production and helping maintain the cells that produce it. A strong mucus barrier allows the immune system to observe and manage microbial life at a controlled distance, rather than being forced into repeated close-range conflict. It is easier to remain calm when the walls are intact. This calm matters because inflammation is useful only in the right amount. Acute inflammation is part of a healthy immune response to infection or injury. It brings immune cells to the scene, increases blood flow, and helps contain damage. But chronic or excessive inflammation wears tissue down, disrupts barrier function, alters metabolism, and can contribute to disease. The microbiome helps reduce the likelihood of this chronic overactivation by shaping a more stable immune environment. When the beneficial community is strong, the immune system often behaves with more balance. When the microbiome is disturbed, depleted, or less diverse, immune regulation may weaken and inflammatory signals may rise. This becomes especially apparent in germ-free animal studies where animals are raised without any microbes at all. These studies, while highly artificial, have taught researchers a great deal about the role of microbiota in immune development. Animals raised in germ-free conditions often show underdeveloped immune tissues, reduced numbers of certain immune cells, altered antibody production, and other signs that the immune system has not matured normally. In other words, without microbial exposure, the immune system is not fully trained. It has missed part of its education. Now, humans are not and should not be germ-free, so this is not a model of health. It is a way of showing that microbial life is deeply involved in immune development from the very beginning. Early life is particularly important in this regard. During infancy and childhood, the immune system is learning rapidly, and the microbiome is changing rapidly as well. Birth method, feeding, antibiotic exposure, environment, illness, and diet all influence which microbes are present and when. These early microbial encounters help shape immune tolerance and responsiveness in lasting ways. Researchers have long been interested in whether disruptions during this period might influence the later risk of allergies, asthma, autoimmune conditions, or inflammatory disease. The picture is complicated, as biology usually is, but the broad idea is compelling. A well-trained immune system may depend in part on meeting the right microbial teachers at the right times. This does not mean dirt is magic, or that every childhood scrape is a sacred immunological ritual. It does mean that the immune system evolved in a world full of microbes, and some of those microbial exposures are part of normal development. Excessive sterility, unnecessary antibiotic use, highly restricted microbial environments, or repeated disruptions of the microbiome may alter the educational signals the immune system receives. The body expects a certain amount of microbial company. Without it, or with the wrong patterns of it, immune regulation may not develop as smoothly. At the same time, the trainers do not only teach tolerance, they also help prime the immune system to respond effectively to true threats. Beneficial bacteria can stimulate the production of antimicrobial peptides by intestinal cells. They can enhance barrier repair after injury. They can shape the readiness of innate immune responses so that when a real pathogen arrives, the body is better prepared. In this way, the microbiome helps the immune system avoid two opposite mistakes. It reduces the risk of overreacting to harmless things and helps maintain the capacity to react appropriately to harmful ones. Wisdom, after all, is not simply knowing when not to fight. It is also knowing when the fight is truly necessary. This balancing act is especially important in relation to inflammatory bowel conditions and other immune-mediated diseases. Although these illnesses are complex and involve genetics, environment, immune signaling, and many other factors, changes in the microbiome are often part of the picture. Reduced diversity, lower abundance of certain beneficial bacteria, and altered metabolite production may contribute to a more inflammatory state. When the trainers are fewer, quieter, or replaced by less helpful organisms, the immune system may receive a very different education. Barrier function may weaken, tolerance may decline, inflammation may become more persistent. The body begins to interpret ordinary conditions through a more suspicious and reactive lens. This also has implications beyond the gut. Immune balance in the intestine can influence the wider immune tone of the body. Signals generated in the gut, including microbial metabolites and immune mediators, can affect distant tissues and systemic inflammation. This is one reason the microbiome has been linked, cautiously but increasingly, to conditions that seem at first glance unrelated to digestion. Skin conditions, metabolic disease, mood-related states, and autoimmune processes may all be influenced, directly or indirectly, by gut-immune interactions. The trainers work locally, but the lessons may be heard elsewhere. Of course, it is important to avoid turning the microbiome into a universal explanation for every human problem from eczema to heartbreak. The gut is influential, but it is not omnipotent. Still, when it comes to immune function, its role is too important to dismiss. A healthy microbiome supports a well-regulated immune system. A disrupted one may make balanced immune behavior harder to maintain. That is a substantial contribution by any measure. Certain microbial species have become especially interesting to researchers because of their apparent immune effects. Bacteroides fragilis, as mentioned earlier, has molecules that can promote immune balance. Faecalibacterium prosnitzi is often associated with lower inflammatory states, perhaps in part because of its butyrate production and other anti-inflammatory influences. Akkermansia muciniphila, known for its relationship with the mucus layer, may also be involved in barrier and immune interactions. Various Clostridia contribute to regulatory T-cell development. Bifidobacteria, important especially in infancy, interact with immune pathways and help shape early tolerance. None of these organisms deserves to be treated as a lone superhero, but together they illustrate how varied the training staff can be. Diet again becomes central here, because the immune supporting functions of the microbiome depend on feeding the right community. Fiber supports fermenters that make short-chain fatty acids. Polyphenols from fruits, vegetables, tea, cocoa, and other plant foods may also influence microbial balance in ways that support immune health. Fermented foods may contribute useful microbes or microbial products. A diet rich in varied plant foods tends to support diversity and the metabolic activities associated with better regulation. A chronically low-fiber, highly processed diet may do the opposite, favouring a microbial pattern that is less supportive of tolerance and barrier health. The immune system listens in part to the chemistry produced by the microbial community, and the community is listening in part to the menu. Antibiotics, illness, and prolonged stress can interrupt this training. When beneficial bacteria are reduced, short-chain fatty acid production may fall, barrier support may weaken, and immune calm may be harder to maintain. Recovery is often possible, but it may take time. This is why health is so often about repeated ordinary support rather than dramatic rescue. Feed the trainers, protect the barrier, avoid unnecessary disruption when you can. The immune system does not need perfection. It needs a stable and informative environment in which to do its work. There is something deeply reassuring about this relationship. The immune system is not left to figure everything out alone. It is shaped by companionship. Tiny organisms living in the gut help show it what normal life looks like. They help define the background against which danger can be recognized. Without them, the body may become more confused, more reactive, or less prepared. With them, especially when they are diverse and well supported, the immune system often behaves with greater discrimination and steadiness. And perhaps that is one of the loveliest truths hidden inside the intestine. The body learns safety not only through shields and walls, but through peaceful contact, repeated exposure, and chemical conversation. The trainers teach by being present. They teach by fermenting fiber, by supporting mucus, by calming inflammatory pathways, by encouraging regulatory cells, and by helping the immune system understand that life includes constant contact with harmless others. That lesson is not weakness. It is one of the foundations of health. So while the day outside may have been noisy, demanding, or uncertain, inside the gut there is likely still a patient lesson being repeated in molecules and cells. Harmless things are being tolerated, threats are being watched, inflammation is being restrained where it does not belong. The body is learning once again how to live with company. And as that quiet training continues, another conversation is unfolding as well, because some of the same microbes shaping immunity also help make chemicals and signals that reach beyond the gut and begin to influence mood, sleep, appetite, and the strange, subtle dialogue between the intestine and the brain. By the time we reach this part of the story, the gut is no longer just a digestive tube full of helpful workers and careful defenders. It has become something much more interesting and much more personal. It is now a place of signals, a place where bacteria do not simply break down food, guard territory, and help train the immune system, but also release chemicals and influence pathways that reach far beyond the intestine itself. This is where the subject begins to feel almost strange, because it forces us to admit something that older biology often kept neatly separated. The gut and the brain are not distant strangers. They're in regular contact. They send messages back and forth all day and all night, and some of the organisms living in the gut help shape those messages in ways that can influence mood, stress, appetite, sleep, and the general tone of how the body feels. This connection is often called the gut-brain axis, which sounds very official and slightly mechanical, as though there ought to be a polished metal lever hidden somewhere behind the ribs. In reality, it is not one axis so much as a whole network of communication routes. The gut talks to the brain through nerves, through immune signals, through hormones, and through microbial products that enter circulation or influence the cells lining the intestine. The brain talks back through stress responses, changes in gut movement, shifts in secretion, appetite changes, and signals sent through the autonomic nervous system. This means the relationship is not one way. The brain influences the gut, and the gut influences the brain. Anyone who has ever felt nausea during stress, lost their appetite during grief, or experienced the urgent digestive effects of a very bad piece of news already knows this in a practical sense. What is newer is our growing appreciation that the bacteria living in the gut are involved in this conversation too. Now it is important not to become overexcited and start imagining that your intestinal microbes are secretly composing your thoughts, choosing your favourite songs, or plotting to make you text someone you absolutely should not text. The brain remains the brain. Your personality is not simply a bacterial committee in a warm room, but the microbiome can influence the chemical environment in which the nervous system operates, and that matters. It matters because mood, sleep, stress, and appetite are not created in isolation. They are shaped by hormones, inflammatory signals, metabolites, nerve activity, and countless other physical processes. If gut bacteria can change some of those processes, then they can influence the conditions under which the brain does its work. One of the most discussed links between the gut and the brain is the vagus nerve. This nerve is a major communication line between the brain and many organs, including the digestive tract. It carries information in both directions. Signals from the gut travel upward to the brain, and instructions from the brain travel downward to the gut. It helps regulate heart rate, digestion, and the general balance between calmer parasympathetic activity and the more alert state associated with stress. Some microbial signals appear able to influence vagal pathways, either directly or indirectly, by affecting the cells lining the gut or the chemical environment around them. In other words, the bacteria do not have tiny telephones, but they may still manage to affect the messages being sent along one of the body's most important nerve routes. There have been animal studies in which certain probiotics appear to influence stress-like behavior or anxiety-like behavior, and some of those effects were reduced when the vagus nerve was interrupted. This does not mean one spoonful of cultured milk turns a worried human into a philosopher calmly watching the rain. But it does suggest that microbial activity in the gut can affect signaling pathways that reach the brain. The vagus nerve is one of the most plausible routes for that effect. It is part of the reason the gut is sometimes called a second brain, though that phrase is often used a little too casually. The gut has its own nervous system, the enteric nervous system, and it communicates richly with the brain. But this is still a partnership, not a replacement. Your intestines are not preparing a quiet administrative takeover. Another major area of interest is neurotransmitter-related chemistry. Gut bacteria can produce or influence molecules associated with brain signaling, including serotonin, GABA, dopamine-related compounds, and others. This is where public discussion often becomes a little too eager. People hear that bacteria can affect serotonin and conclude that happiness is just a question of feeding the right yogurt. Biology is not that cooperative. Still, the link is real enough to deserve attention. Serotonin is a good example. People usually think of serotonin as a brain chemical linked with mood, and it is certainly involved in central nervous system function. But a very large amount of the body's serotonin is actually produced in the gut, particularly by enterochromaffin cells in the intestinal lining. Gut microbes can influence this process. They can affect how much serotonin is produced in the gut, how precursor molecules such as tryptophan are handled, and how signaling through the gut changes in response. Serotonin in the gut is heavily involved in digestion, motility, and communication with the nervous system. It does not simply flow from the intestine into the brain like a cheerful parcel delivery, because the blood-brain barrier and other physiological boundaries complicate that picture. But changes in gut serotonin signaling can still affect the broader gut-brain conversation. Tryptophan, the amino acid needed to make serotonin, is another important piece of the puzzle. Gut bacteria can influence how tryptophan is metabolized. Some of it may be directed towards serotonin production, some toward other pathways, including the kynurenine pathway, which is linked to immune activity and can influence brain-related outcomes. The microbiome affects this balance through inflammation, metabolite production, and interactions with intestinal cells. So when people talk about gut bacteria and serotonin, the real story is less about bacteria directly pumping joy into the bloodstream, and more about microbes influencing the metabolic and inflammatory systems that shape serotonin-related biology. Then there is GABA, gamma-aminobutyric acid, one of the main calming neurotransmitters in the nervous system. Some bacteria, including certain lactobacillus and bifidobacterium species, can produce GABA or influence pathways connected to it. Again, caution matters. GABA produced in the gut does not simply leap into the brain in a neat cinematic moment and extinguish worry like a switch, but microbial effects on GABA-related signaling may still matter through vagal pathways, immune regulation, and local gut-brain communication. The point is not that bacteria directly duplicate the work of the brain. It is that they contribute chemical signals that can influence the body systems involved in stress and calm. Dopamine-related pathways are also part of this larger story. Dopamine is deeply involved in motivation, reward, movement, and anticipation. Certain gut microbes can produce dopamine-related compounds or influence the availability of precursors. As with serotonin and GABA, the relationship is indirect and complicated. It is not accurate to imagine your microbiome as a tiny jazz band deciding how enthusiastic you feel about folding laundry. But it's accurate to say that microbial metabolism influences the body's chemical environment in ways that may affect behavior, stress responses, and energy regulation. Short-chain fatty acids return here as well, because they seem to be everywhere useful in the gut. When bacteria ferment fiber and produce compounds such as butyrate, acetate, and propionate, the effects do not stop at the intestinal lining. These molecules can influence immune balance, inflammation, and even brain-related processes. Butyrate, in particular, has drawn attention because it can affect gene expression and may have neuroactive or neuroprotective effects through indirect pathways. Acetate and propionate may also influence appetite-regulating hormones and energy metabolism. This matters because the brain is highly responsive to the body's inflammatory and metabolic state. A calmer gut with better barrier function and lower chronic inflammation may create better conditions for stable mood and clearer signaling. Inflammation is one of the most important links between the microbiome and the brain. Chronic low-grade inflammation can affect mood, fatigue, sleep quality, and cognitive function. It can alter how neurotransmitter systems behave and how stress is processed. A healthy microbiome tends to support barrier integrity and reduce unnecessary inflammation. A disturbed microbiome may contribute to the opposite. If the gut barrier becomes less effective and inflammatory signals rise, the consequences may not stay politely confined to the intestine. Cytokines and other immune mediators can influence the brain and the body's general sense of well-being. This does not mean every bad mood begins in the colon. It does mean the gut can contribute to the inflammatory background against which mood and mental state are experienced. This helps explain why digestive disorders and mood symptoms often travel together. People with chronic gut issues frequently report anxiety, low mood, sleep disruption, or mental fog. Some of that comes from the obvious burden of discomfort, unpredictability, pain, and fatigue. Living with digestive distress is unpleasant and draining, and that alone would affect mood. But there may also be deeper physiological links involving inflammation, nerve signaling, altered microbial populations, and stress pathways. The relationship goes in both directions. Stress can worsen gut symptoms. Gut imbalance can worsen stress-related symptoms. The loop can become self-reinforcing, which is one reason care for the gut and care for the nervous system often need to happen together rather than as separate projects. Sleep is another area where this relationship becomes especially interesting. The microbiome appears to influence sleep through several possible routes, including immune signaling, circadian rhythms, neurotransmitter-related chemistry, and the metabolism of compounds such as tryptophan. The body's sleep-wake rhythms are governed by the circadian system, and gut microbes appear to follow daily rhythms too. Their populations and metabolic activity can shift across the day depending on feeding times, light-driven physiology, and hormonal patterns. In turn, microbial activity may influence the body's timing systems through metabolites and signaling pathways. Disrupted sleep can alter the microbiome, and changes in the microbiome may contribute to poorer sleep. It is yet another example of a two-way relationship. When sleep is poor for long stretches, the gut often feels it. Appetite regulation changes, food choices may worsen, stress hormones rise, inflammation can increase, gut permeability may shift, microbial balance may change. At the same time, a disturbed gut may influence sleep quality through discomfort, inflammation, altered serotonin pathways, or stress-related signaling. This means the relationship between the microbiome and sleep is not some quaint scientific footnote. It is part of the body's larger system of daily rhythm and recovery. if your sleep has ever gone badly wrong after a period of stress, illness, travel, or eating patterns that made your gut distinctly unhappy, then you have already met this system in person. Appetite and satiety are also shaped by microbial chemistry. Certain gut bacteria influence the production of hormones such as GLP-1 and peptide YY, which are involved in fullness and appetite regulation. Short-chain fatty acids can play a role here too. Microbial fermentation may help signal that a meal has had enough effect, that the body has received useful material, and that it can begin shifting away from active hunger. Again, this does not mean bacteria are solely responsible for appetite. Human eating behavior is influenced by emotion, habit, sleep, stress, reward, social setting, and availability, not just by biology. But microbial activity is part of the hormonal background that makes hunger and fullness feel the way they do. This can become especially relevant when diets are poor over time. A low-fiber diet may reduce the bacteria that produce beneficial metabolites and support balanced appetite signaling. Highly processed diets may shift microbial composition in ways that affect inflammation and satiety. Poor sleep may increase cravings and alter the microbiome further. The result can be a loop in which gut signals, reward pathways, and eating habits all start pushing in the same unfortunate direction. It is not a simple story of willpower. It is a web of signals, and microbes are among the messengers woven into it. Stress hormones also interact with the microbiome. When the body is stressed, cortisol and other stress-related signals can affect gut movement, secretions, barrier function, and immune tone. These changes alter the environment the microbes live in. Some bacteria may grow differently under these conditions. Some beneficial functions may weaken. At the same time, microbial changes may influence how strongly the body responds to stress. Animal studies have suggested that the presence or absence of certain microbes can alter stress reactivity itself. Germ-free animals, for example, often show exaggerated stress responses compared with animals with a normal microbiome, though translating that neatly into human life requires caution. Still, the larger principle seems sound. The microbiome helps shape the body's baseline setting for stress. There is something quietly astonishing in that. It means that the body's sense of strain or calm may be influenced, in part, by the state of an ecosystem in the gut. Not determined by it, not imprisoned by it, but influenced by it. The old idea that mind and body are separate enough to be discussed as if they live in different countries becomes harder to defend the more one studies these systems. The brain feels what the body is doing. The gut responds to what the brain is doing. And the microbes in the gut help shape both sides of the exchange. This is why the research around mood disorders, anxiety, and the microbiome has attracted so much attention. Some studies have found differences in microbial composition associated with depression or anxiety. Some interventions involving probiotics, prebiotics, or diet have shown modest effects in certain people. There is genuine promise here, but also a great deal of complexity. Human lives are messy. Mood is shaped by trauma, relationships, sleep, stress, illness, hormones, social conditions, thought patterns, and countless other variables. No honest scientist should suggest that sadness is simply a microbial shortage, or that anxiety can always be fixed with fermented cabbage and a better breakfast. Yet it would be equally wrong to pretend the gut is irrelevant. The microbiome is part of the environment in which mood happens. Prebiotics, the fibers and compounds that feed beneficial microbes, are interesting here because they may influence mood and sleep indirectly, by supporting the bacteria that produce calming and anti-inflammatory metabolites. Probiotics, live microbes that may confer benefits in certain settings, have also been studied for their effects on mood and stress. Some strains have shown intriguing results, especially among lactobacillus and bifidobacterium species. The term psychobiotics has even emerged to describe microbes or microbial supporting compounds that may influence mental state. It is an appealing term, though it can sometimes sound a bit too much like a brand name for a future cereal. Still, the the concept has scientific weight. Some microbes do appear capable of influencing pathways linked to mood and cognition. Food patterns remain central. Diets rich in diverse plant foods, fermented foods, and minimally processed ingredients tend to support a more varied and resilient microbiome. That in turn may support better gut barrier function, lower inflammation, and a healthier flow of metabolites involved in gut-brain communication. A narrow, ultra-processed diet may do the opposite. Sleep, movement, stress management, and recovery matter too, because the microbiome responds to the broader rhythms of life. These are not glamorous conclusions, but they are trustworthy ones. The things that help the microbiome are often the same things that help the nervous system more generally. Biology has a way of rewarding consistency more than cleverness. One of the gentlest truths in all this is that the gut and brain are never really alone with their own business. They are in contact constantly, and much of that contact happens beneath awareness. You may notice a stressful meeting making your stomach tight. You may notice poor sleep changing your appetite. You may notice digestive discomfort making you feel more irritable than usual. But beneath those obvious experiences, countless smaller signals are moving all the time. Bacteria are fermenting fibers, cells are releasing hormones, immune mediators are rising or falling, nerves are carrying updates, the brain is reading the body, and the body is reading the gut. So while the world outside grows quieter and the day begins to loosen, there may still be a long and subtle exchange continuing inside you. Microbes are shaping metabolites, the gut lining is sending messages, the vagus nerve is carrying information upward, sleep rhythms are being nudged by chemistry, inflammation, and habit. Mood is unfolding within a body that is never just one organ speaking for itself. The chemical messengers of the gut do not replace thought, memory, personality, or choice, but they help create the biological weather in which those things are lived, and that weather can be steadier or more turbulent depending on how well the gut ecosystem is supported. When beneficial microbes are fed, when the barrier is healthy, when inflammation stays controlled, and when daily rhythms are not constantly being pushed into confusion, the messages sent from the gut to the brain may be calmer, clearer, and less disruptive. That does not solve every human difficulty, but it does matter. The mind rests in a body, and the body is listening to its microbes. From there, it is only a short step to another vital part of the story, because many of the same bacterial signals that affect mood and sleep also help regulate something more physical and more immediate inside the intestine itself. They help decide whether the gut lining stays strong, whether inflammation remains controlled, and whether the inner boundary between the body and the world holds together the way it should. If the chemical messengers help explain how the gut can influence mood, sleep, and the brain, then the next part of the story brings us back to the intestine itself, to the place where so much of this invisible work is happening. Because for all the talk of signals and metabolites and bacterial communities, none of it works properly for long unless the lining of the gut stays intact and the fires of inflammation remain under reasonable control. This is where the balancers come in. These are the bacteria that help maintain order at the level of tissue, barrier, and immune tone. They support the cells that line the intestine, help preserve the mucus layer, influence how tightly the cells are joined together, and produce compounds that make unnecessary inflammation less likely. They do not remove the need for the immune system. They do not turn the gut into a perfectly peaceful paradise in which nothing ever goes wrong. But they help keep the system from tipping too far into damage, irritation, and leakiness. They help the gut remain a place of controlled exchange rather than a place where the border begins to fray. That border is one of the most impressive pieces of biology in the body. The intestine is responsible for absorbing nutrients, water, and useful compounds from food, which means it cannot be sealed shut like a vault. It has to be open enough to let the right things through. At the same time, it has to keep back bacteria, toxins, partially digested material, and all the other things that should remain inside the gut rather than wandering into the bloodstream like badly behaved guests. This is a difficult job. The intestinal lining is thin. in many places only one cell layer thick, and yet it performs this balancing act day and night with astonishing reliability when things are going well. That reliability depends on several layers of protection working together. There are the epithelial cells themselves, there are tight junctions between those cells, there is the mucus layer sitting over the surface, there are immune cells nearby, and there is the microbiome, helping to shape the whole environment in ways that support stability. When people talk casually about gut health, they often use the phrase gut lining without quite explaining what it means. The lining is made up of epithelial cells arranged in a continuous sheet along the intestine. These cells are replaced constantly because life in the gut is demanding and the tissue needs renewal. Between them are tight junction proteins, structures that regulate what can pass between one cell and another. If those junctions are functioning well, the barrier remains selective. Nutrients can be absorbed in controlled ways, while harmful material is limited. If the barrier becomes disrupted, substances that would normally stay within the gut can cross more easily, and that can provoke immune reactions and inflammation. The point is not that the intestine should be sealed like plastic wrap. It is that it should remain well regulated. The mucus layer adds another level of protection. In the colon especially, mucus forms a crucial barrier between the dense population of gut bacteria and the epithelial cells underneath. It is not simply slime in the crude and disappointing sense of the word. It is a carefully maintained biological layer made largely from mucins, complex proteins produced by specialized cells called goblet cells. This mucus helps keep most bacteria at a safe distance from the tissue. It also provides a habitat of sorts for certain microbes that interact with it in controlled ways. When the system is balanced, the mucus layer acts like a soft shield, keeping the microbial crowd from pressing directly against the wall. The balancers help maintain both the epithelial barrier and the mucus system. They do this partly through the substances they produce. Short-chain fatty acids are especially important again, particularly butyrate. Butyrate is one of the main products of fiber fermentation by certain gut bacteria, and it serves as an important fuel source for colon cells. This alone makes it valuable, because healthy colon cells help maintain the barrier. Butyrate also appears to influence inflammation, gene expression, and tight junction integrity. In other words, it does not merely feed the lining, it helps regulate how the lining behaves. This is one reason bacteria such as Faecalibacterium prausnitzii, Roseburia species, and certain Eubacterium species are so often discussed in microbiome research. These are among the bacteria associated with butyrate production, and their presence is often linked with healthier gut environments. Faecalibacterium prausnitzii, in particular, has drawn attention because lower levels of it have been observed in some inflammatory gut conditions. It is not the sole answer to everything, and no bacterium deserves to be turned into a miracle mascot, but it is one of the clearer examples of a microbial resident whose activities appear strongly tied to a calmer, better supported intestinal environment. Butyrate is not acting alone. Acetate and propionate matter too, and together the short-chain fatty acids form a chemical bridge between diet, microbes, and tissue health. When beneficial fermenters are well fed, especially by diverse plant fibers, these compounds are produced in greater amounts. They help maintain a lower inflammatory tone in the intestine and support the barrier that keeps the contents of the gut where they belong. A person eating a diet rich in varied plant foods is therefore doing more than promoting regular digestion. They are also supporting microbial pathways that help keep the lining strong and the immune system less reactive. This has profound consequences because inflammation is useful only when it is controlled. Acute inflammation is part of normal defense. If a pathogen invades or tissue is injured, inflammatory signals recruit immune cells, increase blood flow, and help contain the problem. Without inflammation, the body would be dangerously defenseless. But chronic low-grade inflammation in the gut is another matter. It can damage tissue, disrupt tight junctions, alter microbial composition, reduce comfort, impair absorption, and set the stage for further imbalance. Too much inflammation can begin to wear down the very surface it was meant to protect. That is where the balancers do some of their most valuable work. They help keep the immune response from becoming excessive and self-defeating. Several bacterial groups contribute to this calming effect. Some do so through metabolites such as butyrate. Some influence regulatory T cells and other immune cells in ways that encourage restraint. Some support mucus production and thus reduce unnecessary direct contact between bacteria and the intestinal lining. Akkermansia mucinifera is an interesting example here. It is known for its close relationship with the mucus layer. It feeds on mucin, which at first sounds faintly alarming, as though one has invited a very polite saboteur into the system. But in balanced conditions, Akkermansia appears associated with healthier mucus turnover, better barrier integrity, and improved metabolic markers in some settings. The key, as so often in biology, is context and balance. A microbe interacting with mucus in a stable ecosystem is not the same as unchecked erosion of the barrier. Healthy systems involve renewal as well as protection. Bacteroides species also play balancing roles, though again with the usual microbiological caution that not every member of a large group behaves identically. Some bacteroids help process complex carbohydrates, produce useful metabolites, and interact with immune pathways in ways that support tolerance rather than constant attack. Bacteroides fragilis has received particular attention because of molecules such as polysaccharide A, which seem capable of shaping immune balance and encouraging less inflammatory responses. These effects remind us that balancing the gut is not only about feeding the tissue, it is also about teaching the immune system not to overreact at every opportunity. The phrase gut permeability often appears in this discussion, sometimes with more drama than is necessary. The intestine is always somewhat permeable in the sense that it must allow nutrients and water to cross. The issue is not whether anything passes through. The issue is whether the regulation of that passage remains appropriate. When tight junctions are disrupted or the barrier is otherwise damaged, larger molecules, bacterial products, and inflammatory triggers may cross more easily than they should. This can activate immune responses and contribute to local or systemic inflammation. Popular culture sometimes turns this into a catch-all phrase for every modern discomfort, which is unfortunate because the real biology is already interesting enough without exaggeration. The honest point is simply that barrier integrity matters, and certain gut bacteria help maintain it. One way they do this is by influencing the expression of tight junction proteins. Research suggests that beneficial metabolites, especially butyrate, can help support these junctions and the health of the epithelial cells connected by them. When the microbiome is disrupted, when fibre intake is chronically low, or when inflammation is already high, these supportive signals may weaken. The gut lining then becomes more vulnerable. It is not usually a dramatic moment. It is more often a gradual loss of steadiness, a system becoming less resilient and more reactive over time. Diet has an enormous influence here. A varied fibre-rich diet tends to support bacteria that ferment those fibres into short-chain fatty acids. Polyphenol-rich foods such as berries, cocoa, tea, olives, and many vegetables may also support beneficial microbes and help reduce inflammatory stress. By contrast, diets very low in fibre and high in heavily processed foods can starve beneficial fermenters and shift the microbial balance toward less favourable patterns. Some bacteria, when deprived of dietary fibre, may increasingly rely on mucus-derived carbohydrates, which over the long term can place more strain on the protective mucus layer if the rest of the system is not supporting healthy renewal. This does not mean 1 week of poor eating causes the gut wall to collapse in despair, but repeated patterns matter, and the gut remembers what it is consistently given. Antibiotics can disrupt these balancing functions too. They reduce harmful bacteria when needed, but they may also reduce helpful residents that produce butyrate, support mucus, and calm inflammation. In the aftermath, the gut environment may become more chaotic. Recovery often happens, but it may take time, and not all microbial communities bounce back at the same pace. During that recovery period, the barrier may be less well supported, the immune system more reactive, and the ecological balance more fragile. It is another reminder that health often depends on preserving what already works, not simply reacting once things go wrong. Stress also has a place in this story. Chronic stress can affect gut movement, secretion, immune activity, and blood flow to the digestive tract. Those changes alter the environment in which the microbiome and gut lining exist. Stress may increase inflammatory signaling and make the barrier more vulnerable. At the same time, a disturbed gut can feed back into stress through discomfort, immune activity, and chemical signals reaching the brain. This creates the familiar but deeply frustrating loop in which stress worsens the gut and the gut worsens stress. The balancers help resist this cycle by maintaining a calmer intestinal environment, but if the strain becomes too great for too long, their numbers and functions may decline. Sleep naturally matters too. Poor sleep is associated with higher inflammation, altered appetite regulation, stress hormone changes, and disruptions in microbial rhythms. A chronically sleep-deprived body is not giving the gut ideal conditions in which to maintain barrier integrity and immune balance. This does not mean that every late night immediately tears at the intestinal lining like a tiny scandal in the dark. But repeated sleep disruption is yet another way the broader life of the body influences the local ecology of the gut. The balancers do not work in isolation. They work within the larger context of how a person lives. When these balancing functions weaken, the consequences can be broad. The gut may become more inflamed, more reactive, more uncomfortable. Certain opportunistic microbes may gain ground. The immune system may encounter more microbial fragments and interpret them as threat. Digestion may become less smooth. Signals travelling to the rest of the body may carry more inflammatory weight. Over time, these conditions can contribute to disorders in which the gut feels less like a quiet inner ecosystem, and more like a place constantly on the edge of irritation. Not all inflammation begins in the microbiome, of course. Genetics, infections, medications, stress, autoimmune processes, and many other factors matter. But the microbial contribution to whether inflammation stays controlled or becomes harder to manage is substantial. Inflammatory bowel diseases offer one example of how complex this can become. Conditions such as Crohn's disease and ulcerative colitis involve genetic susceptibility, abnormal immune responses, barrier dysfunction, environmental triggers, and changes in the microbiome. Researchers often observe reduced microbial diversity and lower abundance of certain beneficial butyrate-producing bacteria in these conditions. That does not mean the disease is caused by the absence of one helpful species, like a town descending into disorder because one accountant has taken a holiday. It means the balancing ecosystem is altered, and the loss of that balance is part of the disease process. The barrier, the immune system, and the microbiome are all entangled. Even outside overt disease, a less balanced gut can influence how people feel day to day. Mild but persistent bloating, irregularity, food sensitivity, abdominal discomfort, and a vague sense that the digestive system has become touchy or unreliable may reflect a gut environment that is more inflamed or less well supported than it should be. That does not mean every digestive complaint is a microbiome issue. Human bodies are wonderfully varied and sometimes simply difficult. But it does mean the state of the gut lining and the bacteria that support it are often part of the picture. There is also something deeply practical in the fact that many of the bacteria associated with a calmer, stronger gut lining prefer a diet humans are repeatedly told to eat anyway. They like fibre from legumes, vegetables, fruits, nuts, seeds, and whole grains. They respond well to diversity. They benefit from steady patterns rather than nutritional chaos. The advice is not glamorous. It does not come packaged in a silver pouch promising transcendence by Tuesday. It is the same quiet truth biology keeps offering again and again. Feed the system well, and the system has a better chance of staying stable. That stability is one of the less celebrated forms of health. People often imagine health as energy, brightness, strength, or visible fitness. But at the level of the gut, health is also uneventfulness. It is the barrier quietly doing its job. It is the immune system staying calm when calm is appropriate. It is bacteria fermenting fibres, feeding colon cells, supporting mucus, and reducing the odds that inflammation will drift upward for no good reason. It is not a dramatic triumph. It is a long-running competence. And long-running competence is, in its own way, one of the finest biological achievements. The more one looks at the balances, the more one sees that they are not a separate category, so much as a set of functions woven through many familiar bacteria. The digesters become balancers when their fermentation products help calm inflammation. The protectors become balancers when they help maintain the mucus layer and keep pathogens from provoking immune chaos. The trainers become balancers when they teach immune cells restraint. In the gut, roles overlap. That is how ecosystems work. One organism may perform several useful tasks at once, and the value of the system lies not only in individual species, but in the combined effect of their interactions. So while the body outside seems still, and the evening settles further, there may be a whole set of balancing acts continuing within. Mucus is being renewed, tight junctions are being supported, colon cells are being fed, immune signals are being moderated. Bacterial populations are shaping the tone of the gut, making it more likely that food will be tolerated, microbes will remain where they belong, and inflammation will stay within useful limits rather than spilling into something destructive. It is all wonderfully ordinary and wonderfully essential. And of course, balance can be lost. An ecosystem that helps regulate inflammation can also fall out of order when conditions change enough. Helpful residents may decline, opportunists may bloom, the barrier may weaken, signals that were once calming may become less abundant. What was steady can become unsettled, which leads us naturally into the next part of the story, because not all bacteria remain helpful in all conditions, and sometimes the inner world of the gut begins to tilt toward imbalance, overgrowth, and trouble. For a long time, the inner world of the gut can seem almost beautifully organised. The digesters break down fibre, the vitamin makers contribute useful compounds, the protectors hold the line against invaders, the trainers help shape the immune system, the chemical messengers influence the brain and body, and the balancers keep inflammation and the gut lining from drifting into disorder. It is a deeply cooperative picture, and for much of life it is true enough. But every ecosystem has its risks. A forest can burn, a reef can bleach, a city can fall into disrepair when its routines are disturbed and its balance is broken. The gut is no different. There are times when harmful bacteria, or bacteria that are usually harmless in small numbers, begin to overgrow. The conditions that once kept everything in order weaken. The inner world becomes less stable, less predictable, and less protective. This is where the disruptors enter the story. It is tempting to imagine the disruptors as clear villains, arriving dramatically from outside with small wicked suitcases and very poor intentions. Sometimes that happens. Pathogenic bacteria such as Salmonella, Shigella, Campylobacter, or certain strains of Escherichia coli can enter from contaminated food, water, or exposure and cause acute illness. But often the situation is less theatrical and more unsettling, because disruption can also come from within. Organisms that were present at low levels all along may suddenly gain ground. Species that are not especially dangerous in a balanced gut may become troublesome when the system changes. The problem is not always the arrival of something entirely new. Sometimes it is the loss of the conditions that kept old residents in their proper place. This is one of the central ideas in gut ecology. Health depends not only on having beneficial bacteria present, but on maintaining the balance that keeps harmful ones from dominating. When that balance is disturbed, the whole environment changes. Nutrients may become available in different ways. The mucus layer may weaken. The immune system may become more reactive or less well regulated. The pH of the gut may shift. The production of beneficial metabolites such as butyrate may fall. Competition for space may decrease. In that altered landscape, disruptors find opportunity. Scientists often use the word dysbiosis to describe this state of microbial imbalance. It is a useful word, though it can sound slightly like the name of a stern ancient ruler. Dysbiosis does not refer to one single disease or one precise microbial pattern. It is a broad term for a gut community that has shifted away from healthier balance. There may be lower diversity, fewer beneficial bacteria, more inflammatory species, more opportunists, or a general loss of resilience. Dysbiosis can be a cause, a consequence, or both. It may result from illness, antibiotics, poor diet, chronic stress, infection, inflammation, sleep disruption, aging, or a combination of several of these. Once it begins, it can reinforce itself. That is what makes the disruptors so difficult. They are not always the original cause of the trouble. Sometimes they are what happens when a system already under strain starts to slip. One of the clearest examples is Clostridioides difficile, often called C. difficile or C. diff. This bacterium can live in the gut or be acquired from the environment, and under normal conditions, the surrounding microbiome often helps keep it under control. But when antibiotics wipe out large portions of the resident bacterial community, that control can weaken dramatically. Suddenly nutrients are more available, competition is reduced, and the protective environment begins to open up. C. difficile can then multiply rapidly and produce toxins that damage the lining of the colon, causing diarrhea, inflammation, pain, and sometimes life-threatening illness. It is one of the classic reminders that the microbiome is not decorative. When the protective community is damaged, the consequences can be severe. Antibiotics are an especially important part of this story because they are both essential and disruptive. They save lives. There is no question about that. They are one of the great achievements of medicine, but they also do not always distinguish neatly between harmful bacteria and helpful residents in the gut. A broad-spectrum antibiotic may reduce a dangerous infection, which is exactly what it is supposed to do, but in the process it may also shrink beneficial populations that support colonization resistance, better rate production, barrier integrity, and immune balance. The result can be a gut community that is thinner, less diverse, and more vulnerable. Most people recover well after a course of antibiotics, but the recovery is not always immediate, and in some cases it is incomplete. During that time, disruptors may seize the advantage. Antibiotics are not the only route to imbalance. Diet can slowly reshape the microbial landscape too. A chronically low-fiber, highly processed diet may starve many of the beneficial bacteria that ferment plant fibers and produce short-chain fatty acids. Over time, these useful populations may decline. Other bacteria, some less helpful, may expand under the new conditions. This does not mean every slice of cake causes a microbial coup. The gut is more resilient than that. But repeated patterns matter. When the bacteria that help feed the gut lining and calm inflammation are neglected for long enough, the environment becomes less steady. The disruptors do not need a grand invitation, they only need the loss of obstacles. Inflammation itself can also reshape the gut in ways that favour disruption. When the lining becomes irritated and immune activity rises, the chemical environment of the intestine changes. Oxygen gradients may shift, nutrient availability may change. Compounds released during inflammation can favour the growth of certain bacteria that are better adapted to disturbed conditions. Some members of the Enterobacteriaceae family, for example, may bloom in more inflamed settings. These are not always classic pathogens in the dramatic food poisoning sense. Some are normal or near normal residents at low levels, but when inflammation increases they may gain a competitive advantage. The gut then becomes less like a calm ecosystem, and more like a place where a loud and opportunistic crowd has noticed the supervision is weakening. This is part of what makes disruption so frustrating. It often becomes a loop. A changed microbiome increases inflammation. Increased inflammation further changes the microbiome. Barrier function may weaken, allowing more bacterial products to stimulate the immune system. The immune system responds, which further alters the environment. Symptoms grow, stability declines. The inner world falls out of balance not in one dramatic explosion, but in a series of feedback loops that turn a steady system into a reactive one. When this happens, the symptoms can vary widely. There may be bloating, abdominal discomfort, cramps, irregular bowel movements, diarrhea, constipation, urgency, excessive gas, nausea, or a vague sense that digestion has become touchy and unreliable. Sometimes the signs are obviously disruptive. Sometimes they are subtle enough that people simply start describing their gut as sensitive, unpredictable, or easily upset. That is part of the challenge. Dysbiosis is not one thing, and the consequences of bacterial overgrowth or imbalance do not always look identical from one person to another. A useful example here is small intestinal bacterial overgrowth, often called SIBO. Under ordinary conditions, the small intestine contains far fewer bacteria than the colon. That makes sense because the small intestine is a place of rapid digestion and nutrient absorption, with digestive enzymes, bile, and movement patterns that usually keep bacterial numbers relatively controlled. When bacteria overgrow there, especially microbes that are better suited to the colon, the result can be fermentation occurring in the wrong place and at the wrong stage of digestion. This may lead to bloating, gas, discomfort, altered bowel habits, and poor tolerance of certain foods. The issue is not that all those bacteria are inherently evil. It is that they are in the wrong location or the wrong numbers, disrupting processes that depend on proper timing and placement. A perfectly tolerable neighbour is much less charming at 3 in the morning with a tuber. That point matters beyond SIBO as well. Harmful is not always an absolute label. Context matters. Some bacteria are dangerous pathogens under almost any circumstance. Others are opportunists. In a balanced gut, they stay in the background. In a disturbed gut, they expand and contribute to symptoms, inflammation, and tissue irritation. Even microbes often described as beneficial can become problematic if they end up where they do not belong, or if the broader system around them is already damaged. The gut is not governed by moral categories, it is governed by ecology. There are also bacteria that produce toxins or irritating byproducts when they overgrow. Some species break down proteins into compounds that can be more irritating to the gut in excess, especially when diets are very low in fibre and high in the poorly balanced substrates. Others may influence bile acid metabolism in ways that change the intestinal environment. Bile acids themselves are another important part of this story. Beneficial bacteria help transform bile acids as they move through the intestine, and these transformed bile acids can help regulate microbial growth. When the microbiome is disturbed, bile acid patterns can change in ways that may favour disruptors such as C. difficile. Once again, one lost balance creates another. Travel, infections, and stomach illnesses can also trigger longer-term disruption. An acute infection may clear, but the microbiome may not return to its previous state right away. Some people find that digestion remains altered for weeks or months afterward. The lining may have been irritated. The immune system may remain more reactive. Certain bacterial groups may have declined while others expanded. Post-infectious gut symptoms are a reminder that the end of an obvious illness is not always the end of ecological recovery. The gut may still be rebuilding its internal order, while the rest of life expects normal service to resume immediately. Stress belongs in this story too, not because stress alone inserts harmful bacteria into the body, but because it changes the terrain they live in. Chronic stress affects gut motility, secretion, appetite, sleep, and immune signaling. These changes can alter the microbial environment and reduce the resilience of the community. A stressed body may eat differently, sleep worse, move less, digest less smoothly, and produce a different hormonal and inflammatory pattern. All of that can influence which microbes do well. The disruptors thrive not only on bad luck or bad food, but often on accumulated strain. The gut notices how a person lives. This is one reason the inner world of the gut can feel so sensitive during difficult periods of life. Illness, antibiotics, poor sleep, travel, anxiety, irregular meals and inflammatory conditions can all pile together. The beneficial bacteria may decline. Fermentation of healthy fibres may become less effective. Short-chain fatty acid production may fall. The mucus layer may be less well supported. The immune system may become more irritated. Then opportunists begin to expand. It is not that the gut suddenly becomes hostile without reason. It is that the supporting conditions for balance have eroded, and what rises in their place is often less helpful. Akkermansia, Bacteroides, Faecalibacterium, Roseburia, and various bifidobacteria are often discussed in healthier states because of their ties to fibre metabolism, mucus balance, and lower inflammatory tone. By contrast, disrupted states may show reduced levels of these beneficial groups and increased abundance of more opportunistic bacteria. Some studies have observed greater representation of inflammatory-associated species or reduced microbial diversity in people with inflammatory bowel conditions, metabolic disease, or chronic digestive complaints. The exact pattern differs from one case to another, but the larger lesson is consistent. When the inner world falls out of balance, the community becomes less stable and often less protective. This imbalance can affect the whole body, not just the bowel. When harmful bacteria overgrow or the microbiome becomes less balanced, inflammation may rise, the gut barrier may weaken, and microbial products such as lipopolysaccharides may cross more readily into the body's internal environment. That can influence immune activity more broadly and potentially affect mood, energy, appetite, and overall well-being. The gut does not quietly misbehave in one corner while the rest of the body remains entirely indifferent. The body notices, the brain notices, the immune system notices. The whole system feels less steady. And yet, it is important not to turn the disruptors into a story of doom. The microbiome is dynamic. It can recover. Communities can rebuild. Helpful bacteria can return when conditions improve. This is one reason treatments aimed at restoring balance can be so powerful. In recurrent C. difficile infection, for example, fecal microbiota transplantation has shown how profoundly reintroducing a healthier microbial community can restore colonization resistance and shut down the disruptor's advantage. It is an extraordinary example of ecology used as medicine. Instead of merely attacking the harmful organism again and again, the treatment helps restore the community that makes the gut hard to conquer in the first place. Less dramatic recovery happens every day as well. Diets richer in diverse plant foods can help feed beneficial fermenters. Time after antibiotics can allow populations to regrow. Better sleep and reduced stress can help the gut environment stabilize. Some probiotics may be useful in certain contexts, though they are often more modest in effect than advertisements would like us to believe. Fermented foods may support microbial diversity and function. Regular movement can influence gut transit and metabolic health. Ordinary routines of care often matter more than one perfect supplement or one brief spell of excellent intentions. This is because the disruptors usually do not rise from one single moment. They rise from conditions. If conditions change back in favour of the protectors, digesters, trainers and balancers, the gut may gradually become less hospitable to overgrowth and inflammation. The mucus layer may recover, the barrier may strengthen, the immune system may calm, fermentation may improve, colonization resistance may return. The city can become orderly again, though sometimes slowly and sometimes with setbacks. One of the more difficult parts of this topic is that people often want a clean list of bad bacteria to eliminate. It is understandable. Clear enemies are emotionally satisfying. But the gut does not usually offer such neat simplicity. There are true pathogens, yes, but many disruptors are opportunists or ordinary residents behaving differently under altered conditions. The deeper goal is not always elimination. It is balance. You're not trying to create an empty intestine. That would be disastrous. You're trying to support a resilient ecosystem in which harmful overgrowth is less likely and beneficial functions are strong enough to keep the whole environment steady. That perspective changes the meaning of symptoms. Bloating, irritation, irregularity, food sensitivity, or post-antibiotic instability are not always signs that the gut needs to be purged of all bacteria like a frightened kingdom closing its gates. Often they are signs that the ecology needs support, rebuilding, and better conditions. The disruptors become a problem when the rest of the system has lost too much ground. Rebuilding the rest of the system is often the longer-term answer. It is also a reminder of how much invisible work health normally requires. When the gut is balanced, you do not notice all the things that are not happening. Harmful bacteria are not overgrowing. The immune system is not overreacting. The barrier is not fraying. Toxins are not spreading. Nutrients are still being absorbed. Short-chain fatty acids are still being made. The city is still functioning. Only when the disruptors gain ground do we begin to realize how many quiet agreements had been holding everything together. So tonight, as the body rests and the day recedes, it is worth remembering that the inner world of the gut is not guaranteed peace. It is maintained peace. A living balance, supported by food, sleep, immune signals, microbial competition, and the steady work of organisms that do not ask for attention. When that balance is disturbed, the disruptors can turn the intestine into a more inflamed, more fragile, and more unpredictable place. But even disruption is part of ecology, and ecology can be reshaped. Conditions matter. What the gut is fed matters. How the body lives matters. Antibiotics, illness, stress, and routine all leave traces in that hidden world, and because of that, the story does not end with the disruptors themselves. It moves outward to the larger forces that keep tipping the balance one way or another, because what you eat, how you sleep, how stressed you are, and what medicines you take all help decide which microbes flourish and which ones fade. That larger question is where the story begins to feel especially close to ordinary life, because once you understand that the gut is an ecosystem, the next thing you naturally want to know is what keeps changing it. Why does one person's microbiome look different from another's? Why can the same body feel steady for months and then suddenly become bloated, sensitive, irregular, or just vaguely not quite right? Why do some bacterial groups flourish while others shrink? And the answer, more often than not, lies in the shaping forces of daily life. The microbiome is not fixed in the way your bones are fixed, or your fingerprints are fixed. It is responsive. It shifts with food, stress, antibiotics, sleep, illness, movement, routine, age, and the accumulated patterns of how a person lives. Some changes are temporary, some are deeper, some are helpful, some are disruptive. But all of them remind us that the hidden world inside the gut is not sitting still while life happens around it. It is being shaped by life constantly. Food is perhaps the most obvious shaper, and also the most relentless, because it arrives every day. Every meal is a delivery of substances not only to the human body, but also to the microbial community living inside it. What you eat determines which bacteria are regularly fed, which metabolic pathways are used most often, and what kinds of compounds are produced in the gut afterward. If you eat a diverse range of plant foods, fibers, resistant starches, and polyphenol-rich ingredients, you are likely feeding a wide set of microbes that ferment, cross-feed, and produce short-chain fatty acids and other helpful metabolites. If the diet becomes narrow, low in fiber, and heavy in ultra-processed foods, then a different set of organisms may gain ground, while some beneficial residents slowly lose the resources they depend on. The microbiome is, in this sense, a record of repeated meals. This does not mean every breakfast causes a dramatic microbial revolution by lunchtime. The system is more stable than that, and that stability is part of what makes it useful. But repeated patterns matter enormously. A person who eats legumes, oats, vegetables, fruits, nuts, seeds, whole grains, herbs, and a variety of plant foods week after week is building a different gut environment than a person whose diet rarely contains fiber and relies mostly on refined starches, sugary snacks, processed meats, and packaged convenience foods. One pattern tends to support diversity in fermentation. The other often supports less diversity, less short-chain fatty acid production, and a more fragile balance. Fibre remains one of the most important parts of this story because so many beneficial bacteria depend on it. The digesters, balancers, and many of the bacteria associated with a healthier gut environment do some of their best work when fermentable fibres are available. Soluble fibres, resistant starches, and a wide variety of plant carbohydrates provide fuel for bacteria that help maintain the gut lining, regulate inflammation, and shape immune tone. When those bacteria are repeatedly nourished, they remain active. When they are starved, they may decline, and with them decline some of the functions that made the gut steadier in the first place. Variety matters as much as quantity. Different fibres feed different organisms. One bacterium may thrive on the compounds found in onions and leeks. Another may prefer the resistant starch in legumes or cooled potatoes. Another may make better use of fruit pectins, and another may respond especially well to whole grains. A varied diet therefore helps support a varied microbiome. It is not only a matter of eating more fibre in the abstract, it is a matter of broadening the menu for a whole ecosystem. A monoculture is fragile. An ecosystem with many available niches and many species filling them is usually more resilient. This is true in forests, in reefs, and in the colon. Polyphenols add another layer. These plant compounds found in berries, tea, cocoa, olives, herbs, spices, coffee, and many vegetables are not simply antioxidants in the vague and overused way health culture often describes them. They are also substances that interact with gut microbes. Some bacteria can break them down into smaller bioactive compounds. Some polyphenols appear to favour the growth of certain beneficial species or suppress less helpful ones. This means the chemistry of plant foods feeds the microbiome in more than one way. It is not only the fibre, it is also the wide range of compounds that plants carry with them. A plate rich in colour is often rich in microbial meaning. Fermented foods have their place too, though they should be discussed with calm rather than with the breathless excitement of a person who has just discovered kimchi and would now like to evangelize in the supermarket. Yogurt, kefir, sauerkraut, kimchi, miso, tempeh, and similar foods may introduce live microbes or beneficial fermentation products, depending on the food and how it is prepared. They may not take up permanent residence in the gut in large numbers, but they can still influence the environment, add microbial diversity, and sometimes support digestion or immune function. Fermented foods are not a magical shortcut to microbial perfection, but they are one more example of how food can act as an ecological influence rather than merely a source of calories. The timing of eating can shape the microbiome too. Gut bacteria do not live outside the body's daily rhythms. They respond to when food arrives, how often it arrives, and how those patterns interact with the body's circadian system. Irregular eating schedules, constant snacking, nighttime eating, or disrupted sleep patterns may alter microbial activity and rhythmicity. The body, after all, is built around patterns of feeding and fasting, waking and sleeping, light and dark. The microbiome listens to those patterns. It does not merely react to nutrients, it reacts to timing. A body that eats at wildly inconsistent hours, sleeps poorly, and lives in a constant state of circadian confusion may create a different microbial environment than one with steadier rhythms. Stress is another enormous shaper, and one of the more frustrating ones because it reaches the gut through so many routes at once. When a person is stressed, the brain signals the body to shift resources. Stress hormones rise, gut movement may speed up or slow down, secretions change, appetite changes, sleep often worsens, inflammation may rise, blood flow patterns shift. All of this alters the environment in which gut bacteria live. Stress does not need to directly kill beneficial microbes to change the microbiome. It only needs to change the terrain enough that some microbes start doing better and others start doing worse. This is part of why prolonged stress can make the gut feel so different. digestion becomes more sensitive, bloating may worsen, appetite becomes unpredictable. Some people eat less, some eat more, some crave sugar and refined carbohydrates, some start skipping meals and then overcorrecting later. The microbiome is shaped by all of this. A person under chronic strain is not only living with a stressed nervous system, they're also feeding and regulating a gut ecosystem under different conditions. The gut often reflects what the mind and body have been carrying, even when no one has said that out loud. Poor sleep works in a similar way. When sleep is disrupted, the body's hormonal rhythms shift. Cortisol may remain less well regulated, appetite hormones can change, inflammation can increase, blood sugar handling may worsen, food choices often become less steady. The microbiome responds to all of those factors. Research increasingly suggests that sleep and the gut microbiome influence one another in both directions. A healthy microbiome may support better rhythms and a calmer inflammatory state. Better sleep may support a more stable microbiome. Poor sleep, on the other hand, can become one more force pushing the ecosystem toward imbalance. Again, this does not mean one late night destroys microbial civilization, but repeated poor sleep becomes part of the pattern the gut has to live inside. Antibiotics deserve special attention because they are among the most powerful and abrupt shapers of the microbiome. As already mentioned, antibiotics can save lives. They remain one of the most important tools in medicine, but when they enter the digestive system, they often affect more than the target infection. Depending on the antibiotic, the dose, the duration, and the person's existing microbiome, they may sharply reduce beneficial populations, alter diversity, and open ecological space for opportunists. Some people recover quickly, others recover more slowly, some lose only a little stability, others experience prolonged gut changes afterward. The microbiome often rebounds, but not always identically. An antibiotic course is not just an event happening to one pathogen, it is often a storm moving through a whole microbial city. Repeated antibiotic use can magnify these effects. If the microbiome is disrupted again and again before it fully recovers, the result may be a community that becomes thinner, less diverse, or easier to destabilize. That does not mean antibiotics should be avoided when truly needed. It means they should be used thoughtfully, which good medicine already aims to do. When possible, avoiding unnecessary antibiotic use is a way of protecting the microbiome from disruption it does not need to endure. The same logic applies to certain other medications that can shift gut conditions, including acid suppressants, laxatives, and some anti-inflammatory drugs. These medicines may be necessary and helpful, but they also remind us that the gut ecosystem is highly responsive to chemistry. Illness itself shapes the microbiome too. A stomach bug, food poisoning, a feverish viral week in which meals become toast and survival, a period of inflammation, a chronic condition affecting the intestine, all of these can alter the balance of organisms in the gut. Some bacteria may decline because food intake changes. Others may grow because transit time changes or inflammation rises. After the illness is gone, the microbiome may begin rebuilding, but it does not always snap back instantly. That is why recovery can sometimes feel strangely slow. The body may no longer be acutely sick, but the ecosystem that supports digestion and barrier function is still trying to reorganize itself. Travel changes the microbiome in its own way. New foods, different water, different time zones, poor sleep, altered routines, stress, unfamiliar microbes, and the general indignity of airports all make an impression. Some travel-related changes are temporary and harmless. Some are more disruptive, especially if infection or prolonged irregularity is involved. Even without illness, travel can shift eating patterns and microbial exposures enough that digestion feels different for a while. The microbiome is not offended by movement, but it does notice when the conditions of life suddenly become strange. Exercise and physical activity also help shape the gut environment, though usually more gently than diet or antibiotics. Regular movement appears associated in many studies with healthier microbial diversity and function. especially when combined with a good diet. Some of that may be due to effects on metabolism, inflammation, gut transit time, and stress regulation. Movement helps the body in many ways, and the gut is one of the quieter beneficiaries. Extreme training, however, especially when combined with underfueling or chronic strain, can stress the gut rather than support it. As with so many things in biology, the helpful pattern is often not excess but steadiness. Age reshapes the microbiome as well. The gut community of infancy is not the gut community of childhood. The microbiome of adulthood is not identical to that of older age. As people grow older, dietary variety may decrease, medication use may rise, gut motility may change, immune function shifts, illness becomes more common, and social routines may alter eating patterns. All of these can influence the microbiome. In some older adults, diversity declines. In others, dietary habits and general health help preserve a healthier balance. Aging does not doom the microbiome to collapse, but it does make the ecosystem increasingly dependent on the quality of the conditions it is given. Early life experiences cast long shadows too. Birth method, breastfeeding, antibiotic exposure in infancy, childhood diet, family environment, infections, and the general microbial richness or narrowness of early surroundings all help shape the microbiome's foundations. Later life continues reshaping the system, but the earliest years matter because they are the time when the immune system is being trained and the community is first becoming established. It is another example of how the microbiome is not just biological but biographical. It reflects history as much as chemistry. Social and household life play subtle roles as well. People living together often share aspects of their microbiomes. They eat similar foods, touch the same surfaces, share microbial exposures, and sometimes share pets, which are their own walking ecosystems of great enthusiasm and very little concern for boundaries. A household with varied meals, routine, sleep, and less chronic stress may support a different microbial pattern than one shaped by constant disruption and rushed eating. The gut listens not only to nutrients, but to the rhythm of life those nutrients arrive within. One of the more difficult truths in all of this is that the microbiome often changes through accumulation rather than drama. A single stressful week may not do much. One course of antibiotics may pass with only brief effects. One holiday full of oddly beige food may not leave lasting damage. But months and years of repeated patterns build an inner environment. A diet low in diversity, poor sleep, chronic stress, frequent unnecessary antibiotic use, little movement, and constant schedule disruption can gradually produce a gut ecosystem that is less stable, less diverse, and more prone to inflammatory trouble. By contrast, repeated support can build resilience. Diverse plant foods, regular meals, adequate sleep, routine movement, careful medication use, stress recovery, and time after illness can gradually create the opposite. This is why the microbiome responds best to consistency. It is not usually transformed by one superfood, one supplement, or 1 week of excellent intentions followed by a return to chaos. It is shaped by what happens often. That is both less exciting and more reassuring than people sometimes want. Less exciting because there is rarely a dramatic cure-all. More reassuring because everyday habits genuinely matter. You do not need to be perfect. You need to be somewhat consistent. The bacteria living in your gut are not asking for a moral revolution. They are asking, in effect, for lunch and reasonable working conditions. That phrase may sound flippant, but it captures something important. The microbiome thrives under conditions that are almost embarrassingly sensible. Real food, plant variety, fibre, enough sleep, less unnecessary disruption. A nervous system that is not constantly in crisis if that can be helped. Movement, time, recovery. These are not luxurious ideas, they are ordinary ones, and yet inside the gut, they are powerful shapers. They determine which bacterial groups remain well fed, which metabolites are produced, how well the barrier holds, how calm the immune system stays, and how resistant the gut remains to disruption. It is also worth noticing that the microbiome does not respond to ideals, it responds to actual life. A person may know exactly what would support a healthier gut, and still live in a way that makes it difficult to act on that knowledge. Stress, money, time, illness, work, family duties, grief, fatigue, travel, medications, and access to food all shape what daily life looks like. The microbiome is part of the body, and the body lives in a real life, not in a laboratory or a motivational poster. That matters because health advice should be honest. The shaping forces of the microbiome are biological, yes, but they're also social and practical. What a person can do consistently depends on the life they are actually living. Still, even within ordinary imperfect life, the microbiome remains responsive. Better patterns can help, more variety can help, rest can help, time can help. It is a hopeful kind of biology because it does not require starting over from scratch. Ecosystems recover by degrees, conditions shift, populations rise and fall, helpful functions return when they are supported. The inner world is not frozen, it is always listening. So as the evening quietens and the body settles further into stillness, there may be a whole hidden record of the day being written inside the gut. Meals have fed some microbes and not others. Stress has changed the terrain a little. Movement or stillness has altered the pace of digestion. Sleep, if it comes well, will give the whole system a better chance to regulate and restore. Nothing in the microbiome is separate from life. It is shaped by how the day was lived. And after all of these tiny forces have done their work, what remains is the larger question of what it means to care for such a system at all, to know that inside you is a whole ecosystem, quietly responding to your habits, your rhythms, and the many ordinary choices that make up a human life. And so, after all this wandering through the hidden life of the gut, it feels only right to end not with a dramatic revelation, but with something quieter and, in a way, more comforting. Because what we have really been looking at all along is not just a list of bacteria, not just a set of biological functions, and not just another clever system the body happened to develop. We have been looking at a living ecosystem inside you, one that is active every hour of the day and every hour of the night, one that keeps working whether you notice it or not. Whether you're eating lunch, standing in a queue, staring at the ceiling at 3 a.m, or finally beginning to drift into sleep. That may be one of the strangest and most reassuring truths in the whole subject. You do not have to supervise it. You do not have to remember every name, every genus, every metabolite, every short-chain fatty acid, or every detail of immune signaling. You do not need to lie awake anxiously trying to picture the exact number of bifidobacteria handling your dinner. In fact, that would be a very poor use of bedtime. The important thing is simply to understand that this world exists, that it matters, and that much of what keeps you feeling stable, nourished, defended, and balanced is happening below the level of your awareness. Inside the gut, bacteria are still fermenting fiber from meals you may have forgotten eating. They are still competing for space, preventing less helpful microbes from gaining easy ground. They are still helping maintain the mucus layer and the lining of the intestine. They are still producing small compounds that feed colon cells, influence immune behavior, and help shape the larger chemistry of the body. They are still responding to the patterns of your life, to what you eat, how often you sleep, how stressed you have been, how much rest the body has had, and what disruptions have come and gone. Even now, while the room around you is quiet, that inner work continues. There is something humbling in that. We spend so much of life focused on what we can see and feel directly. We notice the mind when it becomes restless. We notice the body when it hurts. We notice hunger, fullness, fatigue, cramps, comfort, discomfort, energy, and heaviness. But beneath all of that is an enormous amount of maintenance, repair, negotiation, and cooperation we never notice at all. The gut is not silent because it is inactive. It is silent because so much of its best work happens without needing to announce itself. Perhaps that is why the microbiome has captured so much attention in recent years. It changes the way we think about what a human body is. We're used to imagining ourselves as self-contained, one organism, one body, one clear boundary between self and world. And in one sense, of course, that is true. You are you. You're not a loose committee of bacteria in a coat pretending to be a person. But in another sense, the body is much more collaborative than older ways of thinking allowed. It contains whole populations of organisms that help extend what it can do. They digest what you cannot digest by yourself. They help produce compounds you need. They influence the way your immune system learns restraint. They help guard a border that is always under pressure from the outside world. They contribute to signals that may travel far beyond the intestine itself. Life, it turns out, is often more shared than it first appears. That does not make you less human. It makes the human body more interesting. It means that what feels like one single life from the outside is also made possible by layers of cooperation on the inside. It means that health is not just a matter of your own cells doing their jobs in neat isolation. It is also a matter of relationships, conditions, environments, and balances being maintained. That may sound like an odd thing to say about the colon, but it happens to be true, and balance really is the word that keeps returning, because almost every useful thing we have talked about depends on it. A healthy microbiome is not one in which only one species dominates. It is not one in which every bacterium is always helpful in every circumstance. It is not a polished fantasy of perfect order in which no discomfort ever occurs and no disruption ever happens. It is a system in which many groups of organisms coexist in ways that support digestion, barrier function, immune regulation, and metabolic steadiness. Some ferment fibers, some make vitamins, some hold off invaders, some help maintain the lining, some help shape the immune system, some influence signaling that reaches the brain. Their usefulness lies not only in who they are individually, but in how they exist together. That lesson matters because people are naturally drawn to simple stories. One bacterium becomes famous, one yogurt promises transformation, one supplement is spoken about as if it were a diplomatic genius, a repair crew, and a therapist all at once. But the gut rarely works through simple stories. It works through communities, through overlap, through compensation, through networks. One microbe may help feed another. One group may produce a compound that another group turns into something even more useful. A tissue cell may depend on bacterial fermentation products. An immune cell may respond to a bacterial signal. A stretch of mucus may be renewed because the whole environment around it is supportive. The system is resilient not because one hero is doing everything, but because many things are being done at once. There is a quiet comfort in that too. It means the body does not depend on perfection from one source. It depends on enough support from many sources, repeated over time. And that brings us gently back to daily life, because the hidden ecosystem inside you is always listening to the ordinary patterns of how you live. It listens to your meals. It notices when fibre is present and when it is absent. It notices whether variety is common or rare. It responds when sleep becomes shallow or irregular. It responds when stress stretches on too long. It responds to medicines, to illness, to travel, to movement, to stillness, to the difference between being fed and being neglected. The microbiome is not frozen. It is always being shaped, which means it is always, at least to some degree, capable of being supported. That does not mean controlling it with precision. No one can do that, and anyone claiming otherwise is probably trying to sell something in an unreasonably expensive glass bottle. The microbiome is too complex, too individual, and too context-dependent to be mastered by slogans. But it can be cared for. That is a gentler and more realistic idea. You care for it by feeding it regularly, especially with diverse plant foods. You care for it by allowing the body enough sleep that inflammation and stress do not constantly distort the terrain. You care for it by avoiding unnecessary disruption where possible, by recovering after illness, by remembering that health is often built through repetition rather than brilliance. You do not have to micromanage the ecosystem. You have to stop making it unnecessarily hard for the ecosystem to do its work, and perhaps that is one of the nicest things about this whole subject. So much of what helps the microbiome is not extreme. It does not require becoming a different person by sunrise. It does not require memorizing Latin names in the produce aisle, or treating every meal like a biological exam. It asks for habits that are already familiar because they help so many other parts of the body too. Eat real food often enough. Include plant variety. Sleep when you can. Move your body. Recover from stress rather than carrying it endlessly forward. Use medicines wisely. Give the system time to rebuild after it has been strained. These are not dramatic instructions. They are ordinary. But inside the gut, ordinary things can have long and powerful effects. There is something deeply human in that idea. So much of health depends not on single moments of excellence, but on repeated acts of decent care. Not spectacular care, not pure care, just decent care, offered again and again. The microbiome responds to that kind of life. It grows more stable when it is given consistency. It becomes more useful when it is fed the materials it knows how to work with. It becomes more protective when it is not constantly disrupted. The body, in this sense, is not asking for impossible virtue. It is asking for enough support to remain in conversation with itself. When that conversation is going well, you may never think about it. You may simply feel normal. Digestion may be uneventful. The immune system may stay calmer. Hunger and fullness may feel more sensible. Sleep may come a little easier. Mood may feel a little steadier. Not perfect, because no human life is perfect, and no microbiome can solve every sorrow, but steadier. That may be one of the most underappreciated signs of health, not dazzling energy, not endless productivity, but a kind of ordinary steadiness. The body is not complaining. The gut is not demanding attention. The system is doing its work without needing applause. And of course there are times when it does complain. There are times when the balance slips, when antibiotics disrupt things, when stress accumulates, when illness passes through, when food becomes narrow and sleep becomes thin and the gut begins to feel less like a quiet ecosystem and more like a touchy, offended houseguest. That happens. Bodies are not machines made to operate under ideal conditions forever. They are living systems exposed to seasons of strain. But even there, the microbiome offers something hopeful. It is dynamic. It can change. It can rebuild. It can recover when the conditions improve. The inner world is not fragile in the dramatic sense. It is responsive. And responsiveness, though inconvenient at times, is also what makes healing possible. That idea may be worth keeping close. The gut is not only where imbalance happens, it is also where balance can gradually return. A different pattern of meals can matter, better sleep can matter, more time can matter, less strain can matter. Recovery is often slow and unglamorous, but it is real. The ecosystem keeps listening, it keeps adjusting, it keeps trying to find a workable order again. The body, in its own quiet way, is always leaning toward repair when it is given the chance. And maybe that is the thought that fits best at the end of a long day. Not that the body is perfect, not that health can be controlled completely, not that every discomfort has a neat explanation or an easy answer, but that inside you there is still work continuing in a steady, useful direction. Cells are renewing, bacteria are fermenting, signals are being sent, the lining is being maintained, the immune system is negotiating, the body is not waiting passively for morning. It is already occupied with the business of keeping you alive in ways so familiar they become almost invisible. If you are lying there now, perhaps a little sleepier than when we began, it may be comforting to remember that rest is not emptiness. Sleep is not a pause in life. It is a condition in which much of life continues differently. The brain changes its rhythm. Hormones shift. Repair becomes more prominent. Immune activity changes. And in the gut, the ecosystem keeps going. The microbes do not clock out just because you do. They continue their small chemical exchanges and their endless practical labour in the dark. It is not sentimental to say that this is a kind of companionship. They are not conscious companions in the human sense, of course. They are not rooting for you or wishing you pleasant dreams, but they are part of the biological world that keeps holding you together while you rest. That can make the body feel a little less lonely. Not romantic, not mystical, just less lonely. You are not a sealed object drifting through life untouched by other forms of living matter. You are part of a network of exchanges, within and without. The food you ate came from living systems. The bacteria in your gut belong to living systems. The body itself is a living system shaped by relationships. Even in stillness, even in sleep, life within you continues as a collaboration. So perhaps tonight there is no need to force a grand ending onto any of this. It is enough to let the thought settle gently that inside you is a whole hidden ecology, working quietly, changing with you, helping you more than you usually realize. It has been with you through ordinary meals and difficult weeks, through stress and recovery, through childhood, illness, routine and rest. It has never been static. It has always been adapting, responding, rebuilding, and doing the strange, practical work of turning food into chemistry and chemistry into support. And as you begin to let the day go, that work is still going on. Somewhere in the colon, fibre is still being fermented. Somewhere along the gut lining, cells are still drawing on the fuel made for them. Somewhere in the mucus layer, bacteria and barrier are still keeping their careful distance. Somewhere in the immune system, restraint is still being taught. Somewhere in the body's wider chemical conversation, signals from the gut are still being folded into the larger experience of being alive. None of it needs your attention right now. It only needs time, and the body's already giving it that. So let the room grow quiet around you. Let the thoughts of the day loosen a little. Let the pressure to manage everything ease, at least for tonight. There is a whole world inside you that knows how to keep working while you sleep, and it has been doing so all along.